मंच एक खुला मंच है, आए जब भी चाहे। किशोर मंच एक खुला मंच है, आए जब भी चाहे। हर किशोर को मिलेगा अवसर, खुलती जाए राहें। आए जब भी चाहे, आए जब भी चाहे, आए जब भी चाहे, आए जब भी चाहे। हो कला सामाजिक ज्ञान, भाषा गणित या हो विज्ञान, कठिन नहीं है विषय कोई भी, करो जरा सा ध्यान। हो कला सामाजिक ज्ञान, भाषा गणित या हो विज्ञान, कठिन नहीं है विषय कोई भी, करो जरा सा ध्यान।
ये है
किशोर मंच।
Hello students, today we are going to discuss the topics how the gene expression is regulated. Under this topic we are going to study about the meaning of gene expression, mechanism of gene expression and an example of gene expression, that is lac operon. Now look into the picture. Now what the picture is trying to illustrate. Now you can see DNA strands, then you can see the RNA formed on DNA, and then you can see the third process, that is protein synthesis. So all the three processes are there: DNA to DNA, that is replication, DNA to RNA, that is transcription, and from RNA to protein, that is translation. And ultimately you can see the formation of polypeptide. What does this picture signifies? That at some places you can see that the DNA is regulating the gene expression, that is the formation of proteins. So that means the information in DNA, it leads to the synthesis of protein. So there is a direct relation between gene and protein. Now when we go into the mechanism of gene expression, then we see that one gene, the gene which is represented by DNA, that forms ribosomal RNA, messenger RNA and tRNA, and that leads to one polypeptide. These polypeptide, it affects the organelle, it affects the enzyme which is proteinaceous and the biochemical reactions taking place in body. So now let us understand what is the meaning of regulation of gene expression. In a simple term we can say that it means that what mechanism stimulate or inhibit expression of gene as required in the cell. That means there is a mechanism to control the amount of a particular protein at a particular time in a cell as per the requirement of the cell. Now in eukaryotes, the gene expression is regulated at 4 different levels. The one is transcriptional level, that is during the formation of primary transcript, processing level, that is at the splicing level, then transport of mRNA from nucleus to cytoplasm, and at the last at the translational level. Now let us understand the lac operon. Now these lac operon, these were proposed by Jacob and Monod in 1961. They worked on E. coli. Now what is the meaning of operon? It means a unit. Now why it is called an inducible operon? Because it is only in the presence of lactose the operon function. Now let us study the different components of lac operon. It consists of a regulator gene, a promoter gene, operator gene and structural genes. There are 3 structural genes as you can see in the picture. The structural gene for beta galactosidase, the structural gene for beta galactoside permease and the structural gene for beta galactoside transacetylase. Now, let us see the different functions of each component. The regulator gene, it controls the operator gene. It produces repressor protein, stops transcription of structural genes by turning off operator gene. The promoter gene, it is located near operator gene. It has 2 functions. The first function, it binds RNA polymerase and second, it determines template DNA for mRNA transcription. The structural genes, it produce mRNA enzymes. The 3 genes are LEC-Z, LEC-Y and LEC-A. The LEC-Z codes for beta galactosidase, LEC-Y codes for lactose permease and LEC-A codes for transacetylase. The operator gene, it controls the structural genes. Now, why it is called the lac operon? So, in the lac operon they studied the breakdown of lactose into galactose and glucose. The lactose is a disaccharide. The lactose, when absent, there is no transcription, but when lactose present, the transcription takes place. So, the regulation of gene expression in this particular example, lac operon, it takes place in 2 steps. 2 conditions are there. The first is the switch off condition. Now, let us study what happens when the operon is switched off. That means the structural genes are not transcribed. In the switch off condition, the regulator gene, it produces a repressor protein. Now, these repressor protein go and binds at the operator site and it prevents the function of RNA polymerase so that the 3 structural genes are not transcribed. So, this condition is called switch off. Now, let us see what happens when the lactose is present in the medium. So, when lactose is present in the medium, it becomes a switch on condition. How does it take place? Now, you can see that in the presence of lactose, the repressor, it binds to the lactose and it makes the repressor inactive so that the repressor in no longer binds to the operator. The operator is free, the RNA polymerase function and transcribes the 3 structural genes and the respective enzymes are produced. So, what is the inference we get from here? That the genes are expressed when the substrate is present. Here the substrate is the lactose and that is how it is regulating the protein synthesis. Now, let us see what are the advantages of gene regulation. The first very important advantage is it is economical as it saves energy. The cell metabolism adjusted to environmental changes and the housekeeping genes, for example, the repressor protein, they are always in the switch on position. Now, let us start with the first point, how it is economical, because the operon will take place only when the cell is required of that product. There are certain genes in the cells which are constantly formed. They are constantly expressed, they form the proteins and these genes are called housekeeping genes. They are very necessary for the cell to survive. And one of the example is the repressor protein that we have seen in the lac operon. Now, let us have the summary of the topic that we have discussed. The formation of protein from RNA is called translation. The process involves activation of amino acids, charging of tRNA, initiation, elongation and termination of polypeptide chain. Ribosomes move 5' to 3' on mRNA. Gene expression means information in DNA is used to synthesize a functional protein. Lac operon is an example of inducible operon, function only when induced by a substrate. Now, you have a small quiz, try this. Where does translation occurs in a cell? How activation of amino acids take place? What is the direction of movement of mRNA? Name the stop codons. What do you mean by gene expression? Name the components of lac operon. Why lac operon is called inducible operon? What are housekeeping genes? And give one example. In the next discussion, we are going to learn about Human Genome Project, DNA fingerprinting. Thank you. Hello students, today we are going to discuss the topics Human Genome Project and DNA fingerprinting. Earlier you have studied about that how DNA forms RNA and RNA forms protein. So till now you have the concept that the genetic constitution of an organism, it lies in the DNA of the organism. So therefore this important project, which is called Human Genome Project, is so important. Now let us study about details of Human Genome Project. Under Human Genome Project, we are going to study about the goals of HGP, the methodologies adopted to study HGP, and the salient features of HGP, and what are the applications of Human Genome Project. The first very important question that strikes our mind is, what is a genome? A genome is all the DNA that is present in an organism. If you look into the picture, the very small nucleus that contain the whole DNA of the organisms, and how the DNA that forms the protein, and the protein affects the phenotype of an organism. Now what is the use of studying HGP? So what we know till now, the DNA has all the information about an organism, and two individuals vary due to differences in DNA sequences. So there is a need to find out the complete DNA sequence in the human. That why there is a biodiversity, why two individuals don't look the same. So the Human Genome Project is a big project. It is also called the mega project. The human genome, as it is known, it contains 3 into 10 raised to the power 6 base pairs, and this project was launched in 1990. Now let us understand some of the important goals of HGP. It is basically to identify 20,000 to 25,000 genes in man. Second, to determine 3 billion chemical base pairs. Number third, to store information in databases. And fourth very important goal is to improve tools for data analysis. All these goals have led to a new branch of biology, what we now today known as bioinformatics. Now let us see the methodologies to study HGP. The first important methodology is express sequence tags, that is to identify genes that expresses RNA. Here we identify only the genes that is expressed. And the second important methodology is sequenced annotation. So that is a blind approach, where the genes are sequenced whether they are coding or non-coding, that it contains both the introns and the exons, and later on the functions are assigned to them. Now what are the steps of DNA sequence? Let us study the steps of the DNA sequence. The first is the DNA is isolated from cell. As you can see in the picture, the cell is homogenized and the DNA is extracted chemically. Then the DNA extracted, it is converted into random small fragments with the help of molecular scissors called restriction antinucleases. The fragmented DNA is then cloned into suitable host using vectors. The host which are generally used is a bacteria or yeast, and the vectors which are used to carry the genome is BAC, that is bacterial artificial chromosome, or YAC, yeast artificial chromosome. If you look into the picture, that shows the picture of E. coli and the yeast, and BAC and the YAC vectors. The next diagram clearly indicates that how the DNA is cloned. Now, here is the donor DNA. The donor DNA is split into the fragments of DNA. It is attached to the vector by the enzyme DNA ligase, and then it was introduced into a host cell, especially the bacterial cells, and when the bacteria replicates, along with that the recombinant DNA molecules also replicate, and in this way you have a clone of DNA molecules. So, the next picture shows that from a small portion of the DNA, so many different copies of the same DNA can be produced, which we call clone. And after that, the DNA is put into the DNA sequencer. As you can see in the picture, that the sample buffer that contains the DNA fragments is passed through a laser, and with the help of fluorescence detector, you can see the different graphs, different colors for the different bases. A red color for the T, a green color, the yellow color for the G, green color for the A. In this way, you have a different color for the different bases and different peaks. So, when the DNA fragments are put into the DNA sequencer, you get the base pairs. Now, let us see what are the applications of this to understanding the biological system, and second, how genes work together. As you can see in the picture that there is a human diversity project. We all are different, we all are unique. No two individuals are unique. So, with the help of Human Genome Project, it is to be understood that what is the basis for the different biological system operating in the nature. Now, the next important topic that we are going to deal with is the DNA fingerprinting. You must have heard about this particular technique which has been used in the criminal offense. So, now let us study in detail about the different process of DNA fingerprinting. So, we are going to study the meaning of the fingerprinting, the principle behind the technique, and the various process. So, what is the meaning of DNA fingerprinting? Basically, DNA fingerprinting is the DNA profile of an individual. It is based on the principle that the DNA has 3 into 10 raised to the power 6 base pairs which are arranged in a definite sequence. Certain regions are repeated, which are called repetitive DNA. These regions are G to C or A to T rich. These regions show polymorphism, that they do not code for any proteins. These polymorphisms are inheritable from parent to children, and these occur due to mutations. So, the variation is at the genetic level. Now, what do you mean by polymorphism? Polymorphism means at this particular point, which we call repetitive DNA, they show variations in the base pairs, and these base pairs remain constant from parent to offspring. This repetitive DNA is also called satellite DNA. They have VNTRS, that is variable number of tandem repeats. These are specific and these are inheritable. So, as the picture shows that you can see the sequence, how the satellite DNA sequence and how much they are coiled in the DNA. Now, during the process of DNA fingerprinting, let us see that which are the sources from where the DNA fingerprinting is done. The sources are the blood, semen, hair, saliva or tissue bits. Now, let us see what are the steps of the DNA fingerprinting. The first step is the sample collection, that is from the site. The second is the isolation of DNA that we have already studied. Then digestion of DNA, that is by molecular restriction enzymes, then splitting into single strand DNA, then the transfer of DNA fragment to nylon sheath, hybridization with label VNTR probe, and the autoradiography to detect hybridized DNA. So, now let us summarize the various steps of the DNA fingerprinting with the help of the following diagram. This particular technique uses Southern blotting. Now, let us see how the blotting takes place with the help of the following diagram. The DNA is extracted. It is restriction enzyme that produces the fragment. then double-stranded fragments separated by electrophoresis, the denatured to single-stranded fragment, transferred to gel to the membrane, and radioactive label probe in a single strand. They cover the membrane with probe and then incubate. Then the excess probe is washed and then it is visualized by exposure to X-ray film, and the film is developed. The DNA typing pattern appears. At the end of the process, you can find the DNA patterns in the form of bands. So, after understanding the technique of DNA fingerprinting, let us try this and try whether you can find out the biological son and daughter. Now, you have the DNA pattern for the mom and the dad, and there are two daughters D1, D2 and two sons S1 and S2. By looking into the DNA pattern of the mom and dad, try to find out which one is a true daughter and which one is a true son. Now, you can see here the DNA pattern of D1, it matches with the mom and dad. So, that is the D1 is a true daughter. On the other hand, if you see for the son, the S1, the DNA pattern is similar to mom and the dad. So, S1 is the true son. So, let us summarize the topic that we have studied just now. Genome is the total DNA present in an organism. Two individuals are not same due to differences in DNA sequences. Human body has 3 billion base pairs. HGP is a mega project. DNA fingerprinting is DNA profile of an individual. The technique is based on the presence of VNTRs which are inheritable. Wide application in forensic department. Now, check your understanding. Human body has chemical base pairs. What are the two methodologies to study HGP? Polymorphisms are, two options are there, inheritable, not heritable. Polymorphism occur due to repetitive DNA is also called DNA. Digestion of DNA is done by expand VNTRs. So, students, we have finished with the molecular basis of inheritance. In the next discussion, we are going to discuss the chapter evolution. Thank you.
Dear students, today we will discuss about the process of evolution. During the discussion, we will also define and explain the evolution and also the viewpoints of various scientists and their theories about evolution. A basic definition of evolution is that evolution is heritable change in a population through time. Now, it is necessary to break down each of these components of this definition a little bit further in order to better understand what that means. Heritable change with an emphasis on the heritable aspect. Most commonly, we used to refer to DNA, the genetic molecule that is passed on from parents to offspring, and codes for the basic development plan as to how our bodies develop through life. The second component of this notion of a population. Change, evolutionary change is not my change as an individual throughout my life. It is the change of the population that I am part of. We do not think of evolution in terms of the level of individual. We think of evolution as acting on populations. So, how population of individuals have heritable change? And the last notion is this concept of through time. Again, evolution is not my change from birth to death. That is something that we refer to as simply as development. Rather, evolution is how I differ from my parents, or even more, how my children differ from their grandparents, my parents. So, evolution occurs when the scale of generations, our point of focus is generally populations. And the component in which we are really looking at about are these changes which are heritable. Those changes which are passed on from one generation to the next. Now, there is a couple of challenges with this, as we move into understanding of these questions in a fossil record. The first of all is that looking at fossils, we are not necessarily looking at populations. We are looking at individual specimens or perhaps samples of specimens. So, one of the first challenge is translating fossil waste data into population data. This notion of through time also comes into place when we look at fossil record. Fossils come from at times very specific point in time. What? Oftentimes from different points in time, and actually understand exactly how much time separate fossils can be a challenge, but something we certainly need to incorporate into the knowledge that we develop out of a fossil record. The final aspect in this notion of heritable change. When we are looking at the fossil records or the associated geological record, we are not looking at an evidence of DNA for the most part. So, my end of this class will certainly be talking a lot about inherited DNA. Instead, what we are talking about is more of logical change in fossils. Differences between skulls, differences between past cranial anatomy, between one specimen and another. We generally believe that differences in morphology correspond to underlying difference in genetic or heritable change. What? This is an issue that we will also further explore later in this class. What evolution is heritable change in a population through time. The geneticist Theodosius Dobzhansky in 1964 famously wrote, "Nothing in biology makes sense except in the light of evolution." And the field of ecology is no exception to this broadly embraced principle. To study ecology without an understanding of evolutionary theory is to watch a sporting event without first learning the rules, players runs, points are scored, whistle thrills. What the guiding principle underlying these events remain a mystery. With an understanding of the rules, however, even the smallest intricacies of the game can be appreciated, even loved. So, it is with ecology, evolution provides a canon by which we may better understand the interactions of organisms with the environments. In this section, we define evolution as it is understood to modern biology and as it applies to ecology. So, comes to the brief introduction of the evolution. Biological evolution, simply put, is descent with modifications. This definition encompasses small scale evolution, that is, changes in gene frequency in a population from one generation to the next. And large scale evolution means the descent of different species from a common ancestor over many generations. Evolution helps us to understand the history of life. Biological evolution is not simply a matter of change over time. Lots of things change over time. For example, trees lose their leaves, mountain ranges rise and erode. But they are not examples of biological evolution because they do not involve descent through genetic inheritance. The central idea of biological evolution is that all life on earth shares a common ancestor, just as you and your cousins share a common grandmother. Through the process of descent with modification, the common ancestors of life on earth gave rise to the fantastic diversity that we see documented in the fossil record and around us today. Evolution means that we are all descent cousins, humans and oak trees, hummingbirds and whales. Evolutionary biology is the study of history of life forms on earth. What exactly is evolution? To understand the changes in flora and fauna that have occurred over millions of years on earth, we must have an understanding of the context of the origin of life, that is, evolution of earth, evolution of stars, and indeed of the universe itself. What follows is the longest of all the construed and conjectured stories. This is the story of origin of life and evolution of life forms or biodiversity on planet Earth in the context of evolution of Earth and against the background of evolution of universe itself. Now comes to the origin of life. When we are looking at stars on a clear night sky, we are in a way looking back in time. Stellar distances are measured in light years. What we see today is an object whose emitted light started its journey millions of years back and from trillions of kilometers away and reaching our eyes now. However, when we see objects in our immediate surroundings, we see them instantly and hence in the present time. Therefore, when we see stars, we apparently are peeping into the past. The origin of life is considered a unique event in the history of universe. The universe is vast. Relatively speaking, the earth itself is almost only a speck. The universe is very old, almost 20 billion years old. Huge clusters of galaxies comprises the universe. Galaxies contain stars and clouds of gas and dust. Considering the size of universe, Earth is indeed a speck. The Big Bang theory attempts to explain to us the origin of universe. It talks of a singular huge explosion unimaginable in physical terms. The universe expanded and hence the temperature came down. Hydrogen and helium formed some time later. The gases condensed under gravitation and formed the galaxies of the present day universe. In the solar system of the Milky Way galaxy, Earth was supposed to have been formed about 4.5 billion years back. There was no atmosphere on early Earth. Water vapor, methane, carbon dioxide and ammonia released from molten mass covered the surface. The ultraviolet rays from the sun broke up water into hydrogen and oxygen, and the lighter hydrogen gas escaped. Oxygen combined with ammonia and methane to form water, carbon dioxide and others. The ozone layer was formed. As it cooled, the water vapor fell as rain to fill the depressions and form oceans. Life appeared 500 million years after the formation of Earth, that is, almost 4 billion years back. According to the Big Bang model, the universe expanded from an extremely dense and hot state and continue to expand today. Did life came from outer space? Some scientists believe that it came from outside. Early Greek thinkers thought units of life called spores were transferred to different planets including Earth. Panspermia is still a favorite idea for some astronomers. For a long time, it was also believed that life came out of decaying and rotting matter like straw, mud, etc. This was the theory of spontaneous generation. Louis Pasteur, by careful experimentation, demonstrated that life came only from pre-existing life. He showed that in pre-sterilized flask, life did not come from killed yeast, while in another flask open to air, new living organisms arose from killed yeast. Spontaneous generation theory was dismissed once and for all. However, this did not answer how the first life form came on Earth. So, my dear students, today we have discussed about the process of evolution, and in the next chapter we will discuss about the different experiments like Operin and Haldane experiments and Urey and Miller experiments. Thank you. Dear students, today we will describe in detail the O'Brien and Haldane experiment. According to their theory, life evolved in the oceans during a period when the atmosphere was reducing, containing hydrogen gas, water, ammonia, methane and carbon dioxide. What? No free oxygen was there. In the early decades of the 20th century, Alexander O'Brien in 1924 and John Haldane in 1929, before O'Brien's first book was translated into English, independently suggested that if the primitive atmosphere was reducing, means as opposed to oxygen rich atmosphere, and there was an appropriate supply of energy such as lightning or ultraviolet light, then a wide range of organic compounds might be synthesized. O'Brien suggested that the organic compounds could have undergone a series of reactions leading to more and more complex molecules. He proposed that the molecules formed colloid aggregates or conservates in an aqueous environment. The conservates were able to absorb and assimilate organic compounds from the environment in a way reminiscent of metabolism. They would have taken part in evolutionary processes, eventually leading to the first life forms. Haldane's idea about the origin of life were very similar to O'Brien's. Haldane proposed that the primordia sea served as a vast chemical laboratory powered by solar energy. The atmosphere was oxygen free and the combination of carbon dioxide, ammonia and ultraviolet radiation gave rise to a host of organic compounds. The sea became a hot dilute soup containing large populations of organic monomers and polymers. Haldane envisaged that groups of monomers and polymers acquired lipid membranes, and that further developments and eventually led to the first living cells. Haldane coined the term prebiotic soup, and this became a powerful symbol of O'Brien-Haldane view of the origin of life. Now, we will discuss another experiment that shower light on the prebiotic environment to the journey of evolution. This is the Urey and Miller experiment. In 1952, Harold Urey tried to calculate the chemical constituents of the atmosphere of the early Earth. He based his calculations on the widely held view that the early atmosphere was reducing one, and concluded that the main constituents were methane, ammonia, hydrogen gas and water. He suggested that his student Stanley Miller should do an experiment attempting to synthesize organic compounds in such an atmosphere. Miller carried out an experiment in 1953 in which he passed a continuous spark discharge at 60,000 volts through a flask containing the gases identified by Urey along with water. Miller found that after a week, most of the ammonia and much of the methane had been consumed. The main gaseous products were carbon monoxide and nitrogen. In addition, there was an accumulation of dark material in the water. Few of the specific constituents of this could not be identified. What? It was clear that the material included a large range of organic polymers. Analysis of the aqueous solution showed that the following had also been synthesized: 25 amino acids, that is the main ones being glycine, alanine and aspartic acid, several fatty acids, hydroxy acids and amide products. The Urey-Miller experiment was immediately recognized as an important breakthrough in the study of the origin of life. It was received as confirmation that several of the key molecules of life could have been synthesized on the primitive earth in the kind of conditions envisaged by O'Brien and Haldane. These molecules would then have been able to take part in prebiotic chemical processing leading to the origin of life. Since the Urey-Miller experiment, a great deal of effort has been spent investigating probiotic chemistry. It has become apparent that organizing simple molecule into assemblies capable of reproducing and evolving is a far greater task than was generally realized during the excitement that followed the experiment. In addition, the view that the early atmosphere was highly reducing one was challenged towards the end of the 20th century and is no longer the consensus view. Although the significance of specific details of the Miller-Urey for the origin of life may now be in question. It began the new scientific discipline of prebiotic chemistry and has been enormously influential in the development of ideas about the origin of life. In similar experiments, others observed formation of sugars, nitrogen bases, pigments and fats. Analysis of meteorite content also revealed similar compounds, indicating that similar processes are occurring elsewhere in space. With this limited evidence, the first part of the conjecture story, that is chemical evolution, was more or less accepted. We have no idea about how the first self-replicating metabolic capsules of life arose. These capsules reproduce their molecules perhaps. The first cellular form of life did not possibly originate till about 2000 million years ago. These were probably single cells. All life forms were in water environment only. This version of a biogenesis, that is, the first form of life, arose slowly through evolutionary process from non-living molecule, is accepted by majority. However, once formed, how the first cellular form of life could have evolved into the complex biodiversity of today is the fascinating story that will be discussed in coming episodes. So, my dear students, today we have discussed in detail the O'Brien and Haldane experiments that told that the life evolved in the oceans during a period when the temperature was reducing. The similar experiment was also carried out by Urey and Miller, which shows that the atmosphere during the origin of life was also reducing one. The molecule formed colloid aggregates or conservates in an aqueous environment. The conservates were able to absorb and assimilate organic compounds. In the next episode, we will discuss about the evolution of human beings in detail. That is, how do we have evolved from the different time periods. Thank you.
Hello, I am Rajesh Kumar. As in previous lecture we have studied about the significant figures, dimensional analysis and laws of chemical combination. In that we have completed already 2 laws, and the third law in this series is law of multiple proportions. This law was proposed by Dalton in 1803. According to this law, if 2 elements can combine to form more than one compounds, the mass of one element that combine with a fixed mass of the other element are in the ratio of small whole numbers. Example, hydrogen combine with oxygen to form 2 compounds, namely water and hydrogen peroxide. As hydrogen plus oxygen gives water H2O. In this 2 gram of H2 and 16 gram of oxygen just give 18 gram of water. The reaction is H2 plus 1/2 O2 gives H2O. Second, hydrogen plus oxygen give hydrogen peroxide. Reaction as H2 plus half of O2 gives H2O2. In this reaction, 2 gram of hydrogen, 32 gram of oxygen gives 34 gram of hydrogen peroxide. Here the mass of oxygen 16 and 32 gram combined with fixed mass of hydrogen 2 gram bear a simple ratio, that is 16:32 or 1/2. Now the next is Gay-Lussac's law of gaseous volume. This law was given by Gay-Lussac in 1808. He observed that when gases combine or produce in a chemical reaction, they do so in a simple ratio by volume, provided all gases are at same temperature and pressure. Example, combination of hydrogen and oxygen. 2 volume of hydrogen always combine with 1 volume of oxygen to produce 2 volume of steam, that is water vapor. Reaction 2H2 plus O2 gives twice of H2O, that is steam. Just we can say 100 ml of hydrogen plus 50 ml of oxygen give 100 ml of H2O. In this case, in this reaction, we can say that 2 volume of hydrogen is combining with the 1 volume of oxygen and giving 2 volume of water, that is H2O. The ratio between the volumes of reactants and products is 2:1:2. Combination between hydrogen and chlorine. 1 volume of hydrogen and 1 volume of chlorine always combine to form 2 volume of HCl gas, as H2 plus Cl2 gives twice of HCl. In this reaction, 1 volume of hydrogen combine with 1 volume of chlorine and gives 2 volume of HCl. The ratio between the volumes of the reactants and the products in this reaction is simple, 1:1:2. Hence, it illustrates the law of combining volumes. Next is Avogadro law. Avogadro proposed this law in 1811. According to this law, equal volume of all gases under similar conditions of temperature and pressure contain equal number of molecules. Avogadro made a distinction between atoms and molecules, which is quite understandable in present time. Example, 2 volume of hydrogen combine with 1 volume of oxygen to form 2 volume of water vapors, as twice of H2 plus O2 gives twice of H2O, that is steam. In this reaction, 2 volume of hydrogen is combining with the 1 volume of oxygen giving 2 volume of water. Applying Avogadro hypothesis, hydrogen plus oxygen gives water, H2 plus 1/2 O2 gives twice of H2O. 1 molecule combining with the half molecule of oxygen to giving 2 molecule of water. Thus, 1 molecule of water contains half molecule of oxygen, but 1 molecule of water contains 1 atom of oxygen. Hence, half molecule of oxygen is equal to 1 atom of oxygen. 1 molecule of oxygen will be equal to 2 atoms of hydrogen. So, atomicity of oxygen is 2. Next is Dalton's atomic theory. To describe the structure of matter which could explain the experimental facts known at that time about element, compound and mixture, and also the laws of chemical combination. John Dalton in 1808 put forward a theory known as Dalton atomic theory. Dalton published a new system of chemical philosophy in which he proposed the following. Number 1 as matter consists of invisible atoms. Second is all the atoms of a given elements have identical properties including identical mass. Atoms of different elements differ in mass. Third point, compounds are formed when atoms of different elements combine in a fixed ratio. Chemical reaction involve reorganization of atoms, These are neither created nor destroyed in a chemical reaction. Now the next is atomic mass and molecular mass. First is atomic mass. The mass of an atom is very small because atoms are extremely small. Today we have sophisticated techniques such as mass spectroscopy for determining the atomic mass. The present system of atomic mass is based on carbon-12 as the standard has been agreed upon in 1961. In this system, 12 carbon is assigned a mass of exactly 12 atomic mass unit, that is AMU, and mass of all other atoms are given relative to this standard. One atomic mass unit is defined as a mass exactly equal to 1/12 the mass of one carbon 12 atom. Hence, 1 AMU is equal to 1.66056 into 10 to the power minus 24 gram. Example, mass of an atom of hydrogen is equal to 1.6736 into 10 to the power minus 24 gram. In terms of AMU, the mass of hydrogen atom is equal to 1.6736 into 10 to the power minus 24 gram divide 1.66056 into 10 to the power minus 24 gram. So that will equal to 1.0078 AMU, atomic mass unit. Note, AMU, that is atomic mass unit, today has been replaced by a symbol that is U, which is unified mass. Now the next is molecular mass. Molecular mass is the sum of atomic masses of the elements present in a molecule. Molecular mass is obtained by multiplying the atomic mass of each element by the number of its atoms and adding them together. We will take an example. Molecular mass of CH4 methane can be calculated as CH4 contain 1 atom of carbon and 4 atoms of hydrogen. So we will add up the atomic masses of each element like this: 12.011 U, that is a mass of carbon, plus 4 into 1.008 U, that is mass of hydrogen. So the molecular mass of methane will be 16.043 unit. Second one is molecular mass of H2O can be calculated as H2O is equal to, as the atoms of hydrogens are 2. So it will be 2 into 1.008 U plus mass of oxygen 16.00. So the molecular mass of water is 18.02 U. Molecular formula mass. The formula of any substance is used to calculate the formula mass instead of molecular mass. Example, the formula of sodium chloride, that is NaCl. The formula mass of NaCl is equal to, again, atomic mass of sodium plus atomic mass of chlorine, as the atomic mass of sodium is 23.0 U plus atomic mass of chlorine is 35.5 U. So the atomic mass of NaCl will be 58.5 U. Now the next topic is mole concept and molar mass. Atoms and molecules are extremely small in size, and their numbers in even a small amount of any substance is really very large. To handle such large numbers, a unit of similar magnitude is required. Just as we denote 1 dozen for 12 items, gross for 144 items, we use the idea of mole to count entities at the microscopic level: atoms, molecules, particles, electrons, ions. 1 mole is the amount of a substance that contains as many particles as there are atoms in exactly, as 12 gram 0.012 kg of the carbon-12 isotopes. Mole of a substance always contain the same number of entities, no matter what the substance may be. The mass of carbon-12 atom was determined by a mass spectrometer and found to be equal to 1.992648 into 10 to the power minus 23 gram. Knowing that 1 mole of carbon weighs 12 gram, the number of atoms in it equal to 12 gram mole 12 carbon, that is 1.992648 into 10 to the power minus 23 gram divided by 12 carbon atoms. So, it will be 6.0221367 into 10 to the power 23 atoms per mole. This number 6.0221367 into 10 to the power 23 atoms entities in 1 mole is so important. It is known as Avogadro constant, denoted by Na. We can therefore say that 1 mole of hydrogen atoms is equal to 6.0221367 into 10 to the power 23 atoms. The mass of 1 mole of a substance in gram is called its molar mass. Example, molar mass of water is 18.02 per mole. Now, the next is percentage composition. The percentage of each element in a given composition of a sample or substance can be calculated. So, we can check the purity of a given sample by analyzing this percentage composition. The percentage composition of an element can be calculated as mass percent of an element is equal to mass of that element in compound divided by molar mass of that compound and multiplied by 100. Example, water, that is H2O, which contain hydrogen and oxygen. So, we can calculate the percentage of hydrogen and oxygen in this water molecule. Molar mass of water, as we have previously calculated, 18.02 gram. Mass percentage of hydrogen is equal to 2 into 1.008 divided by 18.05 and multiplied by 100 will be 11.18%. Mass percentage of oxygen 16.00 divided by 18.02 and multiplied by 100, that will be 88.97%. Empirical formula and molecular formula. Empirical formula, it represents the simple whole number ratio of various atoms present in a compound. Molecular formula, molecular formula shows the exact number of different types of atoms present in a molecule of a compound. If the mass percent of various elements present in a compound is known, its empirical formula can be determined. Molecular formula can be further be obtained if the molar mass is known. Relation between empirical formula and molecular formula. Molecular formula of a compound is simple whole multiple of its empirical formula. Expressing mathematically, molecular formula is equal to n into empirical formula, where n is an integer such as 1, 2, 3. When n is equal to 1, in this case molecular formula will be equal to empirical formula. N is equal to molecular mass upon empirical formula mass. We will take one example, as a compound contains 4.07% hydrogen, 24.27% carbon and 71.65% chlorine. Its molar mass is 98.96 gram. What are its empirical and molecular formulas? We can calculate it, as we will solve this stepwise. First step, calculation of empirical formula. As we have taken first carbon, element is carbon, percentage of element as given in the question is 24.24. Atomic mass of element is 12. So, we can calculate the moles of carbon here. Moles of element as 24.27 divided by 12 is equal to 2.02 moles. Second element for hydrogen, element is hydrogen, percentage of element as given in question is 4.07. Atomic mass of element is 1. So, we can calculate the number of moles applying as 4.07 divided by 1 is equal to 4.07 moles. Our third element as we have taken in the question, that is chlorine. Element is chlorine. Percentage of element is 71.65. Atomic mass of element is 35.5. So, we can calculate the number of moles as 71.65 divided by 35.5, that is 2.02 moles, as we have calculated the number of moles for these three elements. As in further, simple molar ratio is C, we have calculated 2.02, 2.02 is equal to 1, as we have divided the minimum number of molar mass by as we have get the number of moles. So, for the carbon it will be 2.02 divided by 2.02 is equal to 1. For the hydrogen it will be 4.07 divided by 2.02, that will be equal to 2, and for chlorine 2.02 divided by 2.02, that will be equal 1. So, simple molar ratio will be carbon, hydrogen and chlorine is 1:2:1 as shown. So, the empirical formula is CH2Cl. We have to multiply C with 1, hydrogen with 2, Cl with 1. So, we will get the formula CH2Cl. Now, after first step, the second step, calculation of empirical formula mass. We have get the empirical formula, that is CH2Cl. Mass of carbon is 12, mass of hydrogen is 2 and mass of chlorine is 35.5. So, empirical formula mass will be 49.5. And in third step, calculation value of N. N is equal to molecular mass upon empirical formula mass. Molecular mass as previously given in the question, that is 98.96, and empirical formula mass we have calculated in second step, that is 49.5. So, N is equal to 98.96 divided by 49.5 is equal to 2. So, the value of N is 2. Step fourth, calculation of molecular formula. Molecular formula is equal to N into empirical formula. We have got the value of N is equal to 2 and we have got the empirical formula, that is CH2Cl. So, molecular formula will be 2 into CH2Cl is equal to C2H4Cl2. So, in the next lecture we will study stoichiometry and stoichiometric calculations, limiting reagents. Thank you. Hello, I am Rajesh Kumar. As in previous lecture, we have discussed about mole concept, dimensional analysis, and in this lecture, we are going to study about stoichiometric and stoichiometric calculations. The word stoichiometry is derived from the two Greek words, that is stoichion meaning element and metron meaning measure. Stoichiometry thus deals with the calculation of mass, sometimes volume also, of reactant and products in a chemical reaction. Let us know about what information is available from the balance chemical equation of a given reaction. Example, combustion of methane, a balance equation is given below, like CH4 plus O2 gives twice of CO2 plus twice of H2O. In this reaction, methane, oxygen, carbon dioxide and water all are present in gaseous form as indicated by the letter small g. Here, methane and oxygen called reactants and the carbon dioxide and water called products. All reactants and products are given in the above reaction. It has been shown by the letter G, while in case of solid that is to be shown by S, and in case of liquid it to be indicated by the letter L. The coefficient 2, 4, O2 and H2O are called stoichiometric coefficients. Similarly, the coefficient for methane and CO2 is 1 each. According to the above chemical reaction, one molecule of methane react with 2 molecules of oxygen to give 1 molecule of carbon dioxide and 2 molecule of water. 1 mole of methane react with 2 mole of oxygen to give 1 mole of carbon dioxide and 2 moles of water. As in liquid case, 22.7 liter, that is equal to 1 mole in case of liquids, of methane react with 45.5 liter of oxygen to give 22.7 liter of carbon dioxide and 45.4 liter of water. 16 gram of CH4 react with 32 into 2 gram of oxygen to give 44 gram of carbon dioxide and 2 into 18 gram of H2O. From these relationships, the given data can be converted as follow: mass into moles and moles into number of molecules. These are interchangeable within each other. Density is equal to mass upon volume. Example, calculate the amount of water produced by the combustion of 16 gram of methane. The balance equation for the combustion of methane is CH4 plus O2 gives twice of CO2 plus twice of H2O, as all reactants and products are in gaseous form. As the information can be converted, number 1, 16 gram of CH4 correspond to 1 mole. From the above equation, 1 mole of CH4 gives 2 moles of H2O. 2 moles of water is equal to 2 into 2 plus 16 is equal to 36 gram. Now, the limiting reagents. Many times the reactions are carried out when the reactant are not present in the amount as required by a balanced chemical reaction. In such situation, one reactant is over the other. The reactant which present in lesser amount get consumed. After some time, no reaction take place, whatever be the amount of the other reactant present. Hence, the reactant which get consumed limits the amount of products formed, called limiting reagent. Reactions in solutions. Most of the reactions in laboratories are carried out in solutions. So, it is important to know how the amount of substance is expressed when it is present in the form of a solution. The concentration of the solution or the amount of the substance present in its given volume can be expressed in any of the following ways. Number 1, mass percentage or weight percent. Number 2, mole fraction. Number 3, molarity. Number 4, molality. As the first point, mass percent. The formula is mass percent is mass of solute upon mass of solution into 100. Example, a solution prepared by adding 2 gram of a substance A to 18 gram of water. Calculate the mass percentage of solute. It can be solved as mass of solute is equal to 2 gram, mass of solvent is equal to 18 gram, and mass of the solution, which is the mixture of solute plus solvent, is 18 plus 2 is equal to 20 gram. So, mass percentage of A is equal to mass of A upon mass of solution into 100. It will be 2 gram divided by 20 into 100 is equal to 10%. Mole fraction. It is the ratio of number of moles of a particular component to the total number of moles of the solution. As if a substance A dissolve in substance B and their number of moles are Na and Nb respectively, then the mole fraction of A and B are given as mole fraction of A is equal to number of moles of A upon number of moles of solution, as Na divided by Na plus Nb. Mole fraction of B component will be number of moles of B divided by number of moles of solution, as NB is equal to NB upon Na plus Nb. Molarity. It is defined as the number of moles of the solute in 1 liter of the solution. It is denoted by the letter M. Hence, molarity capital M is equal to number of moles of solute upon volume of the solution in liters. Suppose we have 1 m solution of NaOH, that is sodium hydroxide. 1 m NaOH means 1 mole of NaOH present in 1 liter of the solution. Example, how much volume of concentrated 1 m NaOH solution will be taken which contains 0.2 moles of NaOH can be calculated. 1 mole present in 1 liter or 1000 ml milliliter, then 0.2 mole is present is equal to 1000 ml divided by 1 mole into 0.2, that will be equal to 200 ml. So, 200 ml of 1 m NaOH are taken and the enough water is added to dilute it to make it 1 liter. For such calculations, a general formula is also used, that is M1 into V1 is equal to M2 into V2, where M and V are molarity and volume respectively can be used. We will take example like calculate the molarity of NaOH in the solution prepared by dissolving its 4 gram in enough water to form 250 ml of the solution, such as molarity is equal to number of moles of solute upon volume of the solution in liters. So, in step first, number of moles, mass of NaOH upon molar mass of NaOH, that is 4 upon 40 is equal to 1 by 10 or is equal to 0.1 mole. Step second, applying the formula of molarity, that is 0.1 upon 250 is equal to 0.4 mole per liter. So, the final answer will be 0.4 m. Now, the next is molality. It is defined as the number of moles of solute present in 1 kg of solvent, denoted by the letter small m. Molality small m is equal to number of moles of solute upon mass of solvent in kg, that is kilogram. Example, the density of 3 m solution of NaCl is 1.25 gram per ml. Calculate molality of the solution. In this case, we have taken the 3 mole. So, m is equal to 3 mole per liter. Mass of NaCl solution 3 into 58.5 is equal to 175.5 gram. Mass of 1 liter solution is equal to 1000 into 1.25 is equal to 1250 gram. Mass of water in solution 1250 minus 175.5 is equal to 1074.5 gram. So, molality, number of moles of solute upon mass of the solvent in kg. Number of moles of the solute 3 mole, mass of the solvent in kg is 1074.5 divided by 1000, that is 1.074 kilogram. Applying the formula of molality, m is equal to 3 upon 1.074 is equal to 2.79 mole. With this, we have completed this chapter, that is some basic concept of chemistry. In this chapter, we have studied about the different concept like dimensional analysis, significant figures, mole concept, different laws of chemical combinations and different types of numericals and calculations, that is including molarity and molality. That's all. Thank you.
Hello everyone, I am Preeti Kiran, PGT Chemistry, Kendriya Vidyalaya, AGCR. Dear children, early Indian and Greek philosophers established that matter is made up of atom. The word atom is taken from the Greek word atomios. Atomios means uncuttable or undivisible. But these were mere speculations. There were no scientific proof about it. First of all, Dalton proved scientifically that yes, matter is made up of atoms, that is the last and the ultimate smallest particle. Dear children, today we will study about the structure of atom, Unit 2 of class 11th. Early Greek theories by Democritus and Aristotle. Atom is indivisible, or the atom is made up of 4 elements: earth, fire, water and air. John Dalton also said that atom is the ultimate particle of the matter. Dear children, Dalton theory was able to prove the laws of chemical combination, that is, the law of conservation of mass, law of constant composition and law of multiple proportion. But it failed to explain why when a glass rod or an ebonite rod is wrapped with fur or silk produces electricity. Why electricity is generated on rubbing. The various discharge tube experiments by Maxwell, J.J. Thomson and Chadwick establish that the atom is divisible. There are subatomic particles and the fundamental particles are electron, proton and neutron. Here you can see the electrons and the nucleus containing the protons and the neutrons. Now we will study that how these electrons, protons and neutrons are arranged inside the atom. Here you can see that the nucleus is in the center of an atom and electron is revolving around the nucleus. This is a helium atom. Dear children, what we see today, what we know today, that there is a nucleus inside the atom and the electrons are revolving around it. Various atomic models were proposed. First of all, let's study the various atomic models. Thomson plum pudding model. Thomson suggested that atom is spherical in shape and the positive charge is uniformly distributed and electrons are embedded into it. Then came the Rutherford model. Rutherford suggested that there in the center of an atom there is a nucleus which occupies a very small volume. All the mass of the atom is densely concentrated in the nucleus and the electrons are revolving around the nucleus. But now I am telling you the experiment of the Rutherford which led all these conclusions. Let's see. Rutherford performed the alpha particle scattering experiment. He bombarded alpha particles on a thin gold foil and observed the pattern on the zinc sulfide plate. You can see a radioactive substance in a lead block. Yellow, it is a thin gold foil. You can see the alpha particles coming out of the radioactive substance. Alpha particles are positively charged. You can see the alpha rays hitting the thin gold foil and they are passing out of the thin gold foil and hitting the fluorescent zinc sulfide plate. Rutherford observed that most of the alpha particles they passed straight through the gold foil, but few of the alpha particles experience some deflection through a small angle, and 1 in 20,000 approximately bounced back. It deflected back by an angle of 180 degrees. You can see that most of the alpha particles are going straight. Now you can see a little deflection. That alpha particle is just passing near the nucleus and the positive charges, that is the like charges, they are repelling each other. You can see the deflections. By these observations, Rutherford concluded that the mass of the atom is concentrated in the center of the atom, and that is he named nucleus. Most of the space in the atom is empty and electrons are revolving around the nucleus in the orbits. But dear students, Rutherford model fell into a trouble. There were drawbacks. According to the Maxwell electromagnetic theory, the theory says if a charged particle is accelerated, it emits electromagnetic radiations. And we know that electron is negatively charged. When it is revolving around the nucleus, it will emit radiations and it will follow a spiral path, and ultimately it will fall into the nucleus. And hence, Rutherford model was unable to explain the stability of the atom. And if we consider electron to be stationary, then electron is negatively charged and the nucleus is positively charged. Definitely nucleus will attract the electron towards it and electron will get embedded into the nucleus and ultimately the Thomson's miniature model. Let me show you the spiral path observed by the electron. Niels Bohr tried to remove the drawbacks of the Rutherford model. That time there were two major theories that helped Niels Bohr a lot. The first was that the electromagnetic radiations have the dual behavior, a wave nature and particle nature. And the second one, the atomic spectra can only be explained by quantized energy levels. These two formulations helped Niels Bohr to establish his own model that is known as Niels Bohr atomic model. But before picking up that, let me tell you about atomic numbers and the mass numbers. Dear children, now we know that there are three subatomic particles: electrons, protons and neutrons. The nucleus has the positively charged protons and the neutrons which have no charge. Electrons are negatively charged and they are revolving around the nucleus. The number of the protons is equal to the number of electrons because atom is electrically neutral. The number of the protons is known as the atomic number. Atomic number is represented by symbol Z. Mass number. Mass number is equal to the total number of the neutrons and the protons. And the mass number is represented by symbol capital A. Now let me tell you how to represent the atomic number and mass number. First of all, we write the symbol of the element, and then atomic number is represented by a subscript on the left hand side and the mass number by a superscript on the left hand side. It's 8O16. You can see that 8 is the atomic number of oxygen. It is written on the left hand side as the subscript, and 16 is the mass number of oxygen written as superscript on the left hand side of the oxygen. Isotopes. Dear children, there are atoms of an element which have the same atomic number but the mass number is different. Such atoms with the different mass numbers but same atomic numbers is known as isotopes. There are various elements which exist as isotopes. Among the various isotopes, one of the isotope is radioactive and that is known as a radioisotope. Isobars. These are the atoms with the same mass number but different atomic number. For example, 6C14 and 7N14. These are two different elements, carbon and nitrogen, with different atomic numbers. Atomic number of carbon is 6 and atomic number of nitrogen is 7, but the mass numbers are same, that is 14 and 14. Dear children, today I have told you that atom is divisible and how these electrons, protons and neutrons are distributed in an atom and the various attempts regarding these atomic models. Now we have also studied about isotopes and isobars. I hope this is all very much clear to you. In the next class, we will study about the Neal-Bohr atomic model and the various orbit and orbitals and quantum numbers.
Hello viewers. In our previous lesson, we understood what we mean by relation from a set A to a set B. We saw that we can associate elements of a set A to another set B if we know how they are connected, how they are related. So, Aparna liked carnation, Vasudha likes rose and carnation, and the third person may like all the three flowers. Now, today's lesson is about when does a relation become a function. A relation becomes a function when every element of the set A is related to, is associated to, is mapped to one and only one element of set B. So, Aparna likes rose, Vasudha likes carnation, Kritika also likes carnation. That is allowed. So, one and only one flower from the set B is related to the person Kritika or Vasudha or Aparna. So, Aparna cannot like two flowers, and such a relation is called a function. At the same time, an element of the set B, for example, in this case the sunflower is not associated with any element of the set A. So, sunflower is not the image of any element coming from set A, that is perfectly fine. And so, what do we mean and how do we define what a function is from set A to set B? Here is the definition. A relation F from a set A to a set B is said to be a function if every element of set A has one and only one image in set B. Now, the words that are highlighted here are every. So, every element of set A must be connected, must be mapped to one and only one image in set B. It cannot be associated with more than one element of set B. With these two conditions satisfied, the relation qualifies to be termed as a function. So, if I have a relation F and a relation G defined on set A has 5 elements 1, 2, 3, 4 and 5, and the two relations are defined as list of ordered pairs, F and G both being given. Now, which of these relations is a function? You also have to give reasons. So, if you keep the definition in mind, all elements of set A must be uniquely related to elements of the set B. So, as you can see in the first case, we are looking at 1, 3, 2, 5, 3, 3, 4, 5, 5, 3. No element in the first position is connected to more than one element in the second. So, as you can see in G, 1, 3 and 1, 5 both belong to G, that is 1 is related to 3 and 5 both, and therefore, G is not a function, whereas F relates every element of a set A to one and only one element of A itself, and therefore, F is a function. We also use the metaphor of a function machine to understand what this abstract concept, which is so basic and important as we progress further in maths, especially when it comes to calculus. Functions is a very, very important basic concept. What we see as a function, as a relation, as a rule, can be understood if you think of it as a machine, function machine, which processes something which is put inside it through the input chute, and it processes, converts it and throws out something which we may call as outputs. So, in this figure you can see some spheres being dropped and the machine processes them and churns out cubes of related sizes. In language of our functions, we can also call these inputs to be the variable x, and the function machine, if it represents say the function f, then it throws out quantities which we write as f of x. These f of x is the output, x is the input. So, if f is a function from set x to set y and x comma y belongs to f, then we also write y to be same as f of x. Read it as f of x, where y is called image of x under f and x is called the pre-image of y under f. Symbolically, we represent this function from set x to set y as f colon x arrow y. So, f is a function from x to y. Instead of words, we use these symbols f colon x arrow y to understand and represent this exact statement. So, remember, x is the input, f of x is the output, y is equal to f of x, y is the image, x is the pre-image. When we collect all these inputs together and make a set, then all of these inputs together as a set are referred to as the domain of the function. Range is the set of all outputs. So, this is also important and significant enough to be remembered. So, keep this metaphor in mind as we investigate and understand this concept of functions further. So, in this arrow diagram, we again have an association represented between elements of set A and set B. So, first of all, does this arrow diagram represent a function? Yes, definitely. You can see that each element of set A has only one image in set B. So, the association can be expressed as f is a function from set A to B, and how is the image coming out? Twice the element in the set A plus 1. So, 2 times 2 plus 1 gives me 5, 2 times 5 plus 1 gives me 11 and so on. So, I can express this relationship in a mathematical form as f is a function from A to B such that f of x is equal to 2x plus 1. So, the set builder form of the relation now is coming out in a slightly different format, and here we are looking at that same association which could be expressed in mathematical language expressing the function present between the set A to set B. The domain of the function f is all the inputs. So, it is a collection which is nothing but the set A, whereas the codomain of f is the entire set B including the 1 and 9 which are not connected with any elements of set A, and the range of f is only the outputs, collection of all the outputs, which means 5, 11, 13 and 17. So, range and codomain may not be equal. We saw that earlier as well when we talked about codomain and range of a relation. And do not forget that f is always a relation. So, whatever we had learned earlier stays true even now with an extra condition added in. Before we proceed further, let us also take a look at what we mean by real valued functions and real functions. A function which has either R, that is the set of real number, or one of its subsets as its range is called a real valued function. When the domain of f is also either R or a subset of R, then it is called a real function. Most, in fact, all the functions that we are going to be dealing with are going to be real and real valued functions. In higher classes only you will be able to understand anything beyond the set of real numbers, but for the time being all functions are real functions and resulting in real values only. Now, we are moving on to something very interesting. If you remember, we said that Cartesian product which led to relations is a very important concept because it helps us to visualize the mathematical relationship. And when I have collection of points in 2-dimension system, I can always plot them on the Cartesian system, and when they are plotted, they may form a shape, and that shape is what we call as the graph. So, graph of a function are what we are now interested in, and I would like you to think of graph as the faces of functions. If I know graph of a function, you will be able to say many things about it. Let us start with this. Suppose I have a function f from real numbers to real numbers, f of x is 2x plus 1, and I take few of the ordered pairs in this function and I plot them in the Cartesian system. Then it just looks like random points that have been marked. But now if you look closely, this f, the function, is going to have infinitely many ordered pairs in it, because R is the set of real numbers. So, there are infinitely many ordered pairs that are going to be formed which belongs to R cross R. And if I plot all of them on the Cartesian system, then they are going to trace a locus, a path which is nothing but a straight line, which you also can accept with an understanding that if y was equal to f of x, then y is equal to 2x plus 1 is a linear equation and its graph is nothing but a straight line. Now, if I understand this, then from here we will be able to see also that given a graph, am I able to decide that it represents the graph of a function or not? How do we do that? We use something that is called as a vertical line test. The vertical line test says that a relation is a function if there are no vertical lines that intersect the graph at more than one point. Isn't that interesting? Let us try it out. Suppose I have a graph given here and I take vertical lines at different places. If I mark it down in different places, you see that none of these vertical lines are intersecting the graph at more than one point. And the vertical line test therefore says that this graph is therefore representing a function. What about this one? Here I have a circle now. So, is this representing graph of a function? If I take vertical lines at different places, you will see that they are cutting the graph at more than one point, and therefore, this graph does not represent a function. Similarly, if I take another graph, drawing one line is not enough, because in this case, in this position, this red line is not cutting the graph at more than one point. But this must be true for all vertical lines that can be drawn. So, the moment I take another vertical line at a different position, it cuts the graph at not one, but 3 points, and therefore, this graph again does not represent a function of the variable x. So, it may represent a function, but of another independent variable. Well, we will leave that discussion for another time, but this is not a function of the variable x. Let us now familiarize you with some standard special functions. The first one being the identity function. Identity function is defined as a function from real numbers to real numbers, defined as f of x is equal to x. So, every number gets related to itself. When marked on the graph, it turns out to be a straight line. What is the domain of this function? All values that are coming from set of real numbers can be processed by this function, and the output will be the same as what we have entered, and therefore, domain and range are both real numbers. Another interesting function is the constant function. Again mapped from real numbers to real numbers, f of x is c. C is a constant that can change. So, I can have c to be -2, I can have it to be 4, or I can even have c to be 0. It turns out to be a straight line parallel to the x axis. What is the domain of such a function? Definitely all the real numbers. I can process all real numbers under this function, and therefore, f is defined as we say for all real numbers. So, the domain is all real numbers, but the range, the output will, no matter what x is, will always be the c, the constant. So, the range is a single element c enclosed in the curly brackets, the set containing c. Another interesting and very useful function is the modulus function. Again defined from our real numbers to real numbers, f x is equal to modulus x. Now, in that case, what we are looking at all the positive values being generated. Before we look at the graph, let us see if I can restate this rule. Modulus x can be restated as x when x is greater than or equal to 0, negative of x when x is less than 0. So, you know that modulus of -7 is 7. So, it is negative of -7, right. With that in mind, if I plot first part of this function, that is when x is greater than or equal to 0, I get a line again diagonally inclined at 45-degree angle with the x axis. Negative x will be the line in the second quadrant. Put together, the two constitute the graph of f x is equal to modulus x. So, now from the graph, let us see if you can decide what the domain is. Yes, all real numbers, but what about the range? What are the values of the output? Y. Y in this case remains in the along the positive y axis, and therefore, the domain of f is all real numbers, whereas the range is the set starting from 0 including 0 to infinity, all positive real numbers including 0. Another very important function is the signum function, defined as from real numbers to real numbers, f x as 1 when x is greater than 0, 0 when x is equal to 0, minus 1 when x is less than 0. When we plot it, it comes out in 3 parts by the definition. The domain is all real numbers and the range consists of 3 elements -1, 0 and 1. Greatest integer function is again one of the very significant and something very unusual here. It is defined to be the value of the greatest integer less than or equal to x. So, if I have to ask you to find what is f of 2 and then f of 2.73, they both take the value 2. Greatest integer less than equal to 2.73 is 2. Similarly, f of minus 1.04, the greatest integer less than minus 1.04 is negative 2, and so on. So, the domain of f is all real numbers, but the range is always, the outputs are always integers, and therefore, the range is set of integers. Graph looks very interesting here. What would you call it? A step like function. It takes the formation of steps, and therefore, the greatest integer function is often read as step like function. Try plotting this now on your own on graph paper and see if you do get exactly what I have here on the screen. I hope you have enjoyed today's lesson and you will stay with us as we investigate more on the functions in our next lesson. Hello viewers, in this lesson we are going to investigate the idea of domain and range of a function a little more deeply. Getting back to our metaphor of function as a machine, we understood that all the inputs that can be processed by the function machine are called the domain. That is, domain is set of inputs which can be processed by the function machine, whereas the range is the set of outputs. And so, if f is a function from set x to y and ordered pair x comma y belongs to f, then y is f of x and y is image of x under f. X is also called the pre-image of y under f. Keeping in language of what f being a relation as a collection of ordered pair is, we also define domain to be the set of all first elements of the ordered pairs in the function f from set x to set y. That is, domain is collection of all values of x, whereas range is the set of all second elements, that is the image known as, written as most often as y, in a relation f from a set x to set y. Now, that being the basic definition, are we ready now to look at more complex functions like this one to find the domain and range of the function? So, this is a special function given as square root of 9 minus x squared. What is the domain and range of the function? That is, what are the values over which this f can act and what are the values that this function f produces? In order to understand this, let us take a quick look at a little visualization and understand exactly what this function looks like and how it behaves. Do you know what the graph of square root of 9 minus x squared looks like? No? Then follow me here. This is how you may get the graph of square root of 9 minus x square if you use any graph software, either an online graph calculator, and here I have used GeoGebra. This root 9 minus x square actually is nothing but the graph of a semicircle lying above the x axis. Now, A is any point on this semicircle, and as I move the point A, you will see a red trace being followed on the x axis. That is nothing but the values of the x, that is the first element present on the ordered pair which is following this relation. So, all x coordinates of the point A are being plotted along the x axis, and you can see that in this case that trace did not go beyond -3 and 3 on either sides of the point. So, we are looking at the red trace remains strictly between -3 to 3. How does one get that understanding algebraically? That is, without having this visualization or a software just next to us, how do I get the domain of this function? Let us get back to our algebraic work for that. Now, you know very well that square root of a negative number is not a real number, and we are talking about real functions and real valued functions, and therefore, with that condition we begin our working. If f of x is root of 9 minus x squared, then it is only defined when 9 minus x squared is greater than equal to 0, and from there we find the condition on x. As you can see, x square must be less than equal to 9, and that gives us x lying between -3 and 3, both the endpoints included. That is how we express our domain. Domain of f is therefore the closed interval -3 to 3, and that is exactly what you saw in that visualization earlier. All the values of x were lying enclosed between the interval -3 to 3, starting from -3 stopping at 3, inclusive of these two endpoints. Now, how about the range? The range of this function can be calculated by starting with y written as root of 9 minus x square. Now, square root of 9 minus x squared is always a positive quantity. Square root function is defined only, that is, the outputs of the square root functions are defined to be always the positive values. so that the function or in fact the expression, the connection, the correspondence remains a function. There is a unique number mapped to root of 25. So, root of 25 is not plus and a minus 5, but only as per the definition positive 5. With that in mind, the very first condition on y is that y is greater than or equal to 0. At the same time, if I square both these equations, I will get y square is equal to 9 minus x square, getting a condition on y through x. X squared therefore is equal to 9 minus y squared. What does this tell me? 9 minus y squared must be greater than or equal to 0, because x square is such, and therefore y lies between minus 3 and 3. And therefore, if I put number 1 condition and the second condition in mind, the intersection of the solution on y turns out to be y lying between 0 to 3, 0 and 3 included. And therefore, we say that the range of f is a closed interval 0, 3. Going back to the shape of the function, as I said earlier, graphs are the faces of the function. So, if I know that this function represents a semicircle, then you can see that in this picture the y values cannot be less than 0, cannot be more than 3, and that is what the range of the function is, collection of all the outputs values of y put together as a set. And therefore, when you see this blue mark there, that represents all the values of y that every point on the graph can take, and so range of function is a closed interval 0, 3. So, our next question says find the domain and range of the function f where f of x is equal to 1 by 1 plus x square. Now, what you really need to understand is that the domain consists of all the values of x for which f is defined, f can act on them. In this case, the denominator 1 plus x square can never be 0, right, because when x square plus 1 is 0, it would mean x square is equal to negative 1, which is not going to happen because x square is always positive. And that means that f is defined for all real numbers, and that is what the domain of f would be. And so, we say that since f is defined for all real values of x, the domain of f is set of all real numbers. If you look at the graph of this function, you would find it to be a very interesting shape, and all the values of x that can be traced on the x axis corresponding to every point on the graph covers the entire of the x axis, which is what the set of real numbers are. So, we are looking at the domain of f to be the entire of real numbers. How about the range? Now, I am not going to give you an answer right now, but first look at the graph and see if you are able to estimate the values of y. Can y be negative? No. With the shape right there in front of you, you can definitely say that. Also, 1 plus x square is always positive, and therefore, 1 by 1 plus x square will also be positive, and so the outputs will always be positive real numbers. Will they be ever equal to 0? Can 1 by 1 plus x square be equal to 0? No. So, you have a condition inbuilt in the function's definition itself. So, for the range, if I start with f x equal to 1 by 1 plus x square, then one of the things that I can always do is I can write it to be y equal to 1 by 1 plus x square, and from there find what the condition on x in terms of y would be. So, expressing x in terms of y, I have x square is equal to 1 minus y by y. Now, let us apply the logic. X square is always greater than or equal to 0, and x is a real number. So, the right hand side, that is 1 minus y by y, must always be a real number and must also be greater than or equal to 0. That means y definitely cannot be 0. That is what we saw by the picture as well earlier. Here therefore, the other condition on y would be, since 1 minus y by y is a quotient of two expressions, and that would always be positive if either both the functions, that is expression 1 minus y and y, are positive or both are negative. The common solution therefore, if 1 minus y is greater than or equal to 0 and y is strictly greater than 0, because y could not be 0 as we saw earlier, then the common solution that arises from here is that y must be less than or equal to 1 and y must be greater than 0. The other possibility comes up with y must be greater than or equal to 1 and y is less than 0. Now, is that possible? Can you have y greater than or equal to 1 and less than 0 simultaneously? No. So, the second possibility is not going to happen, and therefore, the condition on y that we get, that is the range that we are looking for the function f, comes out to be same as the interval which is open at 0, but close at 1. Y is greater than 0, but less than or equal to 1. So, if you re-look at the graph of this function, you will understand and appreciate what you have done right now. You have found the values of y which are exactly giving that shape to the curve. The y remains little more than 0, but less than or equal to 1, and that is the significance of the term range of a function. So, both domain and range together decide what the shape of the function is going to be, that is the graph of the function going to be, and vice versa. If you know the graph, you know the domain and range. One more question: find domain and range of the function f where f of x is equal to x plus 1 by 2x plus 3. Now, these are very tricky functions to graph on a graph paper, but we understand that if it is a rational function, it can only be defined when the denominator is not 0, and that is how you begin thinking about the domain. The domain would be for all those values of x for which 2x plus 3 is not equal to 0. That is when x is not equal to minus 3 by 2. And therefore, what is the domain? Domain is all real numbers except the number negative 3 by 2. So, we write it as the difference of real number with the set containing minus 3 by 2. Simple, right? To find the range, very mechanically what you may do is write y equal to x plus 1 by 2x plus 3, cross multiply, collect the terms of x and express x in terms of y. Doing that, you get x is equal to 1 minus 3y by 2y minus 1. Now, apply the same logic as we had earlier. X is a real number other than minus 3 by 2. Therefore, the right hand side must also be a real number. That means, the denominator in the right hand side, which is 2y minus 1, should not be 0. Isn't that so? And therefore, y cannot take value 1 by 2. Other than that, there is no condition on y. So, the range of f turns out to be all real numbers except 1 by 2. So, both domain and range are actually the entire of real numbers except for a particular number. For domain, negative 3 by 2 is never touched on, and for range, half is never attained. So, the output never results in 1 by 2. Does that affect the shape of the graph? Definitely. The graph of this function looks something like this. Maybe you have never tried and plotted such a function earlier. And it is interesting to note that when x is minus 3 by 2, that is, look closely at x is equal to minus 1.5, and you will find that there is no corresponding point or part of the graph at which you will be able to mark a point. Also, since the range did not have half in it, so corresponding to y equal to half, there will be no point on the graph. That means, the graph will not attain the height of half 1 by 2.5, and that is what is so interesting about functions, domain and their ranges. This concept that we have done today is definitely an abstract concept, but if you sit and try some more functions of the similar kind, you will soon start enjoying it. Let us see if you can do that, and I hope it works for you. Good luck.
Hello viewers, today we will discuss some of the world problems involving linear equations. Now what these world problems are? The world problems are practical problems and involve relation between known and unknown. And these world problems are often stated in words and can be mathematically formulated. Let us see one example. It goes like this. In a fraction, if the numerator of a fraction is doubled and denominator is increased by 3, the fraction becomes 5 upon 6. But if the numerator is increased by 3 and denominator is doubled, the fraction becomes 4 over 9. Find the fraction. Now, while doing these problems, you have to take care of some of the things. What are those? First of all, you must read the problem carefully and note down what is given and what is required. After that, denote the unknown quantities by the variables. After reading the problem carefully and denoting the unknowns by variables, translate the statement of the problem step by step into mathematical statement. Later, look for the equal quantities and write the equation by writing appropriate expression. And finally, solve these equations and check whether the solution satisfies the given conditions or not. We have a short while ago narrated one problem. Let us have that problem once again. In a fraction, if numerator is doubled and denominator is increased by 3, the fraction becomes 5 over 6. But if the numerator is increased by 3 and denominator is doubled, the fraction becomes 4 over 9. Find the fraction. Now you know that in a fraction, it consists of numerator and a denominator. And for the fraction which we are talking about, neither the numerator nor denominator is known to me. So what I will do? I will suppose the numerator to be x, and now here x is a variable, and denominator to be y. Now the next condition with this was that if the numerator is doubled, so the fraction becomes x over y. Now when the numerator is doubled, it will become 2 times x. So that on doubling the numerator we get 2x, and denominator is increased by 3. Denominator we have supposed as y. So denominator now becomes, on increasing by 3, becomes y plus 3. So what this fraction becomes then? The fraction now becomes 2x upon y plus 3, and this is equal to 5 upon 6. Now having got this, now we will simplify this statement, this mathematical statement. Now have a look at that mathematical statement once again. This is 2x upon y plus 3 equals 5 upon 6. So on simplification, this will become 6 times 2x equals 5 times y plus 3, and that gives me 12x equals 5y plus 15. Now, transferring this 5y onto left hand side, I get 12x minus 5y equals 15. So, from the first condition I got one equation, and that equation is 12x minus 5y equals 15. Now, there was another condition in the problem, that was when the numerator is increased by 3. Now, what was the numerator we supposed? Numerator we had supposed as x. Now, when it is increased by 3, it will become x plus 3, and denominator is doubled. What was denominator? That was y. So, on doubling the denominator, it becomes 2y. So, therefore, the fraction will become x plus 3 upon 2y equals 4 upon 9. Now, this is an equation involving two variables x and y, which can be simplified as 9 times x plus 3 equals 4 times 2y, and that is equal to 9x plus 27 equals 8y, and on transferring this 8y to one side onto LHS, we get 9x minus 8y equals minus 27. So, now we have two equations from the two conditions, and these equations are 12x minus 5y equals 15 and 9x minus 8y equals minus 27. Now, you have already learnt how to solve them graphically and algebraically. Now, it is always not convenient to solve the equations graphically. So, in most of the cases we follow the method of solving the equations algebraically, but keep in mind, in case in your examination it is asked from you to solve the equations graphically, then you have to solve the equations graphically. Now, let us solve these equations. Here I decide to find the value of y first. So, that means the first equation is 12x minus 5y equals 15 and 9x minus 8y equals minus 27. So, for that I will have to make the coefficients of x same. For that I will multiply the first equation by 3 and second equation by 4. On multiplying by 3 first equation, we will get 36x minus 15y equals 45, and second equation by 4. So, that also makes it 36x minus 32y equals minus 108. Now, since the coefficients of x are same, that is 36 and 36, and both are having the positive sign, what do? We subtract the second equation from this one, and on subtraction the signs will change. This will be to minus, this will change to plus, and this will also change to plus. This x part will cancel out. So, I have 17y equals 153, which gives me the value of y as 9. So, having obtained the value of y, what do then? We have to get the value of x, and to get the value of x, what do? We substitute the value of y in one of the equations. So, let us substitute that value in one of the equations. So, one of the equations is 9x minus 8y equals minus 27, and because we found the value of y to be 9, so that becomes 9x minus 8 times 9 equals minus 27. So, here 9x minus 72 equals minus 27. Transferring this minus 72 onto the right hand side of equality, we get 9x equals minus 27 plus 72, which is 45, and therefore, the value of x comes as 5. Now, recall what was the fraction. In the fraction, we had supposed numerator to be x and denominator to be y, and we have already calculated the value of x and y. So, therefore, our original fraction was x upon y, and that is equal to 5 upon 9. So, the fraction is x upon y equals 5 upon 9. So, here you saw one of the practical problems of mathematics involving fractions. Let us have another problem. The fare from Delhi to Allahabad is 900 rupees for 3 full tickets with reservation charges, and for 4 children below 12 years of age along with reservation charges it is rupees 680. Further, if the fare for a child is half the fare of the full ticket if the child is below 12 years of age, but the reservation charges are same, then what are the reservation charges and the price per ticket? Now, here what are the variables? The one is the fare and the other is the reservation charges. So, what we will assume? Let the fare for the full ticket be x and the reservation charges be y. Now, for 3 full tickets, the fare for 3 full tickets will be 3x and the reservation charges will be 3y. Now, the cost of the ticket has to be added to the reservation charges. So, 3x plus 3y makes it 900. Now, in the second condition, it was provided to us that the fare for a child below 12 years of age is half the fare of the full ticket. So, the fare for half a ticket will be x by 2, and since 4 children are there, so this will be 4 times x by 2, but the reservation charges are same. So, that will be 4 times y equals 680. Now, look at these equations very carefully. In the first equation, we have 3 as the common factor. So, we can divide the first equation by 3, because on dividing a equation, the equation does not change. So, on dividing the first equation by 3, I get x plus y equals 300, and the second equation I will divide by 2. So, that makes it x plus 2y equals 340. So, we got these two equations, and now I need to find out the values of x and y. And how do I go for that? The values of x and y can be found out by solving these two equations simultaneously. See the equations once again. The first equation is x plus y equals 300 and x plus 2y equals 340. From the first equation, I get x equals 300 minus y, and from the second equation, this x plus 2y equals 340, I get x equals to 340 minus 2y. Now, we got the value of x from both the equations. Now, eliminating this variable, we get 300 minus y equals 340 minus 2y, and now transferring the constant terms onto the right hand side by equality and variables onto left hand side, I get minus y plus 2y equals 340 minus 300, and that will give me y equals 40. Now, can you tell me what y was? Y was the reservation charges. Now, I am supposed to find out the cost of the ticket as well. So, for that I will make use of one of the equations. Let me make use of the equation x plus y equals 300. The value of y, which is representing the reservation charges, that has been found out as 40. So, substituting this value, we get x plus 40 equals 300, and therefore, x is equal to 260. And now, you can very well make out that what is the cost for full ticket? The cost for full ticket is rupees 260 and the reservation charges are rupees 40. But since the half ticket, that is the ticket for a child below 12 years of age, is half the fare of the full ticket. So, therefore, the fare for half ticket will be 260 divided by 2 and that is equal to 130. And now a short while ago we had discussed that after you have solved the problem, you must see whether it satisfies the given conditions or not. So, let us see, we have found out the fare for full ticket as 260 and the reservation charges as 40. Now, we had 3 full tickets. So, that means 3 into 260, added to it the reservation charges for 3 tickets. How much that comes to? That is 780 plus 120 and that is equal to 900. So, this is satisfying the first condition. And what was the second condition? 4 tickets, 4 half tickets. So, 4 into 260 by 2, that will be the price of fare for 1 ticket plus the reservation charges for 4 tickets, that is 4 into 40, that will be equal to 520 plus 160 and obviously this total comes out to be 680. So, after having solved this, now we are sure that our calculation is correct, that is the fare for the full ticket was rupees 260 and the reservation charges were rupees 40. So, let us take now another problem which concerns about the area of a rectangle. The question goes like this: if the length of a rectangle is reduced by 5 units and its breadth is increased by 3 units, the area gets reduced by 9 square units. But if the length is increased by 3 units and the breadth is increased by 2 units, its area gets increased by 67 square units. Now, having seen the problem, what do you feel are the unknown quantities over here? The unknown quantities are the length of the rectangle and the breadth of the rectangle. So, what we will assume? We will assume the length to be, we can take it as x or any other variable. Here I will take the length as L and the breadth of the rectangle I will take as B. Now, when the area of the rectangle, when the original length is L and the breadth is B, the area of the rectangle is length times breadth. So, that will be L times B, that is equal to L B. But the first condition was that the length is reduced by 5 units. Now, what is the original length? Original length is L. On reducing it by 5 units, the new length becomes L minus 5 and the breadth is increased by 3 units. What is the original breadth we have supposed? B. Now, it is increased by 3 units. So, increasing means to original breadth I will add 3. And what is happening to area? Area is decreased by 9 square units. So, original area is L times B, because it is decreased by 9 units. So, I will subtract 9. Now, we will equate this new area. The new area will be L minus 5 into B plus 3 equals new area, which is 9 square units less than the original area, that is L times B minus 9. So, having found one equation, we will have to simplify this equation to get a linear equation in L and B. How do we go for that? Let us see that. This is L minus 5 into B plus 3 is equal to L B minus 9. Now, on expanding this, we will get L times B into L times 3, that is plus 3L, minus 5 times B minus 5 times this 15 is equal to LB minus 9. Now, here LB, this quantity and this quantity, they are existing on both sides. So, this we can cancel out. So, we get 3L minus 5B equals minus 9 and minus 15 transfer to the right hand side. This will become plus 15. The first equation we get is 3L minus 5B equals 6. So, this is the first equation we got from the first condition. Now, we had the other condition also. What was that? The other condition was the length is increased by 3 units. On increasing the length by 3 units, we get L plus 3, and increasing the breadth by 2 units, we get B plus 2. The area increases by 67 square units. So, the original area is L times B plus 67, and from here the area of a rectangle is given by length times breadth. New length is L plus 3 times B plus 2 equals LB plus 67. Now, this can also be simplified. Now, when we expand this side, we get LB plus 2L plus 3B and plus 6 equals LB plus 67. Again, LB onto left hand side is positive quantity, and here it is also positive quantity, that can cancel out. So, we get the final equation as 2L plus 3B equals 67 minus 6, and 67 minus 6 is how much? That is 61. So, we got two equations as 3L minus 5B equals 6, and second equation as 2L plus 3B equals 61. Now, we have to eliminate one of the variables, that is one of the unknowns. Now, here I decide to remove B first and evaluate the unknown L first. For that, I will multiply the first equation by 3 and second equation by 5. Can you make out why I am multiplying the first equation by 3 and second equation by 5? Because to eliminate B, I will have to make the coefficients of B in the first equation and in the second equation same. So, multiplying the first equation by 3, I get 9L minus 15B equals 18, and the second equation is 10L plus 15B equals 305. Now, the coefficients of B have been made same, that is minus 15, this is 15 and plus 15, and their signs are opposite. One is minus 15 and other is plus. So, what do? We add these two equations to cancel out B. So, I get 19L equals 323, and from there I will get the value of L as 323 divided by 19, and that is equal to how much? And 7. So, having got the value of length, which was one unknown, I have to get the value of the other variable, and for that what do? I put the value of this length L, value of L, in one of the equations. So, 3 times 17 minus 5B equals 6, and that will give me 3L 51 minus 5B equals 6. Solving it further, I get minus 5B equals 6 minus 51, and that is equal to minus 45, and the value of B then comes to be, the value of B comes out to be 9. So, here we got the value of length as 17 units and that of breadth as 9 square units. Let us have a check for this. How do we go for check? The check is performed by putting the values of length and breadth in one of the conditions. Length we have calculated as 17 and breadth 9. So, what is the original area? The original area becomes 17 times, original area is 17 times 9, that is 153 square units. Now, recall what was the first condition. First condition was the length is reduced by 5 units. So, new length is 17 minus 5 and the breadth was increased by 3 units. So, that means the new breadth will be 9 plus 3 and that is equal to 12 times 12. This is the new area and new area becomes 144 square units. Now, in the problem it was already stated that when length is reduced by 5 units and breadth is increased by 3 units, the area is reduced by 9 square units. Let us see, does it satisfy that condition? The original area is 153. Now, when 153 original area is reduced by 9 square units, we get 144 square units, which is same as the value which I have calculated. So, this is the way we go for the problems concerning the area of a rectangle. Here once again I remind you that we go for the unknowns in the form of variables, that is in the last problem we took the length as L and breadth as B. It is not always necessary to take the unknowns as X and Y. Now, let us see another problem. Now, in this problem this concerns about a classroom. Now, in a class if 3 students sit on each bench, then 6 students are left. But if 4 students sit on each bench, then 2 benches are left. Find the number of students and the benches in the class. Listen to the problem once again. In a class, if 3 students sit on each bench, then 6 students are left. But if 4 students sit on each bench, 2 benches are left vacant. We have to find out the number of students and the number of benches. Now, here what is the condition now? The condition here is that the benches and the students both are not known to us. So, what we will do? We will suppose the number of benches to be x and number of students to be y. Now, if 3 students are sitting on each bench, then the number of students which will sit on x benches will be 3x, but in that case 6 students are left over. 6 students are left over. So, that means if we add to it 6, I get the total number of students. Listen to this once again. If 3 students sit on each bench, that means the total number of students sitting on benches will be 3x, but 6 students are left over. So, that is 3x plus 6 and number of students I have already supposed as y. So, that becomes 3x plus 6 equals y. But if 4 students sit on each bench, 2 benches are left over. So, that means original benches are x and 2 benches are left over, that is new benches will be x minus 2, and because 4 students are sitting on each bench. So, this is 4 times x minus 2. This will be equal to number of students, and on simplifying this I get 4x minus 8 equals y. So, we got 2 equations, the one 3x plus 6 equal to y. Now, here I have equated the number of students to the total number of students supposed, and here also I have equated the number of students to the total number of students. Now, these equations can be solved by the methods which have already been taught to you. So, today viewers, we saw that how the simultaneous equations are applied in word problems, that is practical problems which involve known and unknown quantities. You also do practice for some of these questions.
कल 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 साई साई चिड़ै के बोली, कल 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 साई साई चिड़ै के बोली, गौआ में साँझ हो आए हो, गौआ में साँझ हो आए हो, गौआ में साँझ हो आए। काने पे हर लागे बैलन की जोड़ी, काने पे चले रे किसान लेके कनुआ पे ओढ़ी, चले रे किसान बैलन की घंटी से भरल अनाजबा, बैलन की घंटी से भरल अनाजबा, धरती के दिन आए हो, गौआ में साँझ हो आए हो, गौआ में साँझ हो आए।
तारों ने ली मीठी तान, गूंज उठी स्वर लहरी, घन मंजीरे खनक उठे,
थाप पड़ी जब गहरी।
वाद्य यंत्रों की श्रेणियों में हमने तार वाद्य की बात की,
फूंक मारकर बजाए जाने वाले वाद्यों से मुलाकात की और घन वाद्य के बारे में जाना।
और अब बारी है अवनद्य वाद्यों की, जो चमड़ों से मढ़े हुए साज हैं।
अवनद्य चमड़े से मढ़े हुए ताल वाद्य, समझ गई, जैसे तबला, ढोलक वगैरह वगैरह।
हाँ, ढोल और नगाड़े भी है ना।
हाँ, बिल्कुल तुमने ठीक कहा।
अवनद्य वाद्य, क्या ये ताल वाद्य हैं? पर भैया घन वाद्य भी तो ताल वाद्य ही हैं।
तुमने बहुत अच्छा प्रश्न किया मंजरी, और इन्हीं अवनद्य वाद्यों की विशेष जानकारी के लिए हम तुम्हें तबला मर्मज्ञ और तबला विद्वान पंडित विजय शंकर मिश्र जी से तुम्हारी परिचिति करवाते हैं, जो तुम्हें अवनद्य वाद्यों के बारे में विशेष जानकारी देंगे। पंडित जी, आप इन बच्चों को अवनद्य के बारे में कुछ बताएं।
तुमने बिल्कुल ठीक पूछा मंजरी। घनवाद्य और अवनद्य वाद्य दोनों ही ताल वाद्य हैं, लेकिन दोनों में एक अंतर है। घनवाद्य धातु या लकड़ी के बने हुए होते हैं, जैसे मंजीरा और घंटे आदि, जबकि अवनद्य वाद्यों के बजाने का स्थान चमड़े से मढ़ा हुआ होता है, जैसे तबला, पखावज, ढोलक, नगाड़ा वगैरह वगैरह। हमारे देश में अवनद्य वाद्यों के इतने प्रकार हैं कि हम शायद उन्हें ठीक से गिन भी न पाएँ। अलग अलग प्रकार, अलग अलग आकार, अलग अलग ध्वनियाँ और अलग अलग संगीत शैलियाँ। इन्हें समझने के लिए तुम कोई एक ताल चुन लो तो।
भैया मैं 3 ताल बजाती हूँ।
हाँ ठीक है।
धा, धिन, धिन, धा, धा, धिन, धिन, धा, धा, तिन, तिन, ता, ता, धिन, धिन, धा, धा।
अब ये तबले पे कैसे बजेगा, इसको देखते हैं। एक बात मैं और बताना चाहूँगा आप लोगों को कि हमारे यहाँ अवनद्य वाद्यों के कई प्रकार हैं, लेकिन ये सारे वाद्य एक दूसरे से कहीं न कहीं से जुड़े हुए होते हैं। एक वाद्य के बोलों को दूसरे वाद्यों पर भी बजाया जाता है। इसे देखने के लिए हम तबला और ढोलक का एक प्रयोग देखते हैं। हाँ गौतम, आओ तो जरा। अब हम देखते हैं कि तबला और ढोलक ने एक दूसरे के बोलों को किस तरह अपनाया है। इसके लिए हम कहरवा ताल लेते हैं, जो ढोलक का ताल है। पहले उसको तबले पे सुनते हैं और फिर ढोलक पे। अब इसको सुधीर जरा आप ढोलक पर सुनाएंगे। हमारे यहाँ अवनद्य वाद्यों का प्रचलन अत्यंत प्राचीन काल से रहा है। अनेक पौराणिक ग्रंथों में भी इनका उल्लेख है, लेकिन तब इन्हें तबला या ढोलक नहीं कहा जाता था। तब इन्हें दुंदुभी और पड़व जैसे नामों से बुलाया जाता था। रामायण और महाभारत में भी कई अवनद्य वाद्यों का उल्लेख आया है, जैसे मृदंग, भेरी, पट आदि आदि।
मृदंग, ये नाम तो भैया आज भी प्रचलित है।
हाँ, और मैं ये भी सोच रहा हूँ भैया कि इन वाद्यों से जुड़ी कोई कथा भी आपको जरूर मालूम होगी।
ओह, तो ये बात है, तुम अवनद्य वाद्यों से जुड़ी कोई कहानी सुनना चाहते हो? ठीक है, मैं तुम्हें एक कहानी सुनवाता हूँ।
पुराने समय की बात है। कहा जाता है कि स्वाति नाम के एक ऋषि थे। उनके आश्रम से कुछ दूर एक सरोवर था, जिसमें ढेरों कमल खिलते थे। सरोवर कमल के बड़े बड़े पत्तों से भरा हुआ था। एक दिन ऋषि स्वाति उस सरोवर में स्नान करने गए। एकाएक घने बादल छाने लगे। वर्षा की बूंदें टपाटप पड़ने लगीं। कमल के बड़े बड़े पत्तों पर तेज बूंदें पड़ने से एक विशेष ध्वनि उत्पन्न होने लगी। ऋषि स्वाति उस ध्वनि को सुनकर मुग्ध हो गए। उस ध्वनि को हृदय में बसाए वे अपने आश्रम लौटे और उन्होंने देव शिल्पी विश्वकर्मा की सहायता से एक वाद्य तैयार किया। इस वाद्य को नाम दिया गया प्रणव या दरदुर।
तो ये थी प्राचीन वाद्यों से जुड़ी एक कथा। उपलब्ध प्रमाणों से हमें ज्ञात होता है कि पहले सूखे पेड़ों के मोटे तनों को काटकर उस पर चमड़ा मढ़कर लकड़ी से प्रहार करके उसको बजाया जाता था। गांव-गांव में मुनादी करने के लिए या नृत्य और संगीत का उत्सव मनाने के लिए। आज भी कई जनजातियों में ऐसे ढोल हमें मिलते हैं जिनसे मुनादी कराई जाती है या वो लोग अपना उत्सव इससे मनाते हैं।
हाँ, हमने बहुत सी कहानियों में पढ़ा और सुना भी है नगर नगर ढिंडोरा पीटने की बात।
सुनो सुनो सुनो, छोटे बड़े सभी ध्यान से सुनो।
आप सब शामिल होइए और आनंद उठाइए।
विविध अवनद्य वाद्यों को देखने चलते हैं संग्रहालय। तो आज सुनते हैं तुम्बक नारी, जो कश्मीर का लोक वाद्य है। कैसा आकर्षण है संगीत का।
हम सभी लय, ताल और स्वरों की मधुरता में खो जाते हैं।
हाँ, लय और ताल, और क्या है इनका संबंध, ये जानेंगे अगले कार्यक्रम में।
Water is life. Water is a major constituent of all living matter on the earth. when the rains fail, there is a scarcity of water all around. In some parts of the country, there is continuous drought, whereas in some others, there are floods. Why is it so? There is something wrong somewhere, which results in suffering for all. Let us see how we waste water. Most of our water storage, use and conveyance methods are wasteful. Our canals waste a lot of precious water by seepage through their earthen beds and banks, which results in development of saline and alkaline soils. Continuous sufferings have made us realize that water is precious. We must therefore store it, conserve it, and use it judiciously. With stable side slopes and with banks strong enough to carry the required flow of water safely. A small farmer can also go in for prefabricated pipes or channels for carrying water from the well to the field and thus conserve water for irrigation. He must ensure that there is no leakage at joints. Brick, stone or concrete masonry are commonly used for for lining irrigation channels. Single-layer bricks or stones laid in cement or lime mortar provide virtually waterproof channel lining. Precast structures can prove to be very useful while constructing water channels. Weeds around the lined channels take away part of the water meant for irrigating crops. Remove them as soon as they appear. The ground in its natural state is seldom suited for efficient application of water. Land grading means reshaping the field to a planned grade so that flow of water can be controlled to check soil erosion and provide surface drainage to the field. You are seeing a buck scraper cutting the earth from high patches and depositing it in low patches. This operation is continued till the desired finish is obtained. Land grading gives great benefits to irrigated areas. A properly graded land surface ensures an unobstructed and smooth flow of water into the land without eroding the soil and helps in the uniform distribution of water throughout the field. Due to ploughing and other normal cultivation operations, the smoothness of a graded or leveled field is disturbed. High and low patches develop in some places. Proper shaping of the field can be done by minimal earth movement. This can be done by a bullock-drawn plough or harrow. When irrigation water is released in the field, it should move forward as well as downward. A tractor can also do the job very efficiently. If the field does not have proper smooth shape and has high and low patches, the forward movement of water is restricted. More water accumulates in lower patches, and water may not reach the higher patches. As a result, crop growth is not uniform. To overcome these drawbacks, land shaping becomes necessary. The land which was earlier not fit for growing crops has now been graded, leveled and shaped for efficient irrigation. The water requirements of different crops vary widely, depending on the type of soil. In India, the soils are broadly classified into several groups. The important ones are alluvial soils, laterite or lateritic soils, red soils and black soils. The depth of soil is also important from the point of view of water requirement of different crops. A soil profile will enable the farmer to find out what is there under the topsoil, where the roots of the plants will spread. For surface irrigation of fields, different methods are adopted. Check basin irrigation is the most common method used in India. The field is divided into smaller unit areas. Bunds or ridges are constructed around the areas, forming basins, within which irrigation water can be controlled. The basins are filled to the desired depth, and the water is retained until it infiltrates into the soil. Furrow irrigation is used for all cultivated crops planted in rows, like maize, sorghum, sugarcane, cotton, tobacco and groundnut. The most popular method of irrigation is border irrigation. The field is divided into a number of long parallel strips called borders that are separated by low ridges. Siphon tubes are useful in conveying water over the channel banks into the field. The central feature of border irrigation is to provide an even surface over which the water can flow down the slope with an almost uniform depth. Each strip is irrigated independently. In the sprinkler method of irrigation, water is sprayed into the air and allowed to fall on the ground in a manner resembling rainfall. The spray is produced by the flow of water under pressure through small orifices or nozzles. All the sprinkler irrigation equipment is quite light and easy to carry from one place to another. Assembling the equipment is also simple. Sprinkler irrigation can be used for almost all the crops except rice and jute. By selecting the right size of nozzle, maintaining the desired operating pressure, and keeping proper spacing between sprinklers, the crop will receive efficient and uniform irrigation, which will suit the infiltration rate of the soil. Drip irrigation is one of the latest methods of irrigation. In this method, the water is applied to precisely the root zone. The plants are thus watered frequently with a volume of water appropriate to the level of their consumption. Conventional losses, as by deep percolation, runoff and evaporation, are thus avoided. The system applies water slowly to keep the soil moisture within the desired range for plant growth. Crops like banana, grapes, papaya, guava and other fruit trees and vegetables respond well to drip irrigation. Experiments are in progress to use ordinary plastic tubes for drip irrigation, which will further reduce the cost of having this system in your field. In the areas of water scarcity, plants can be irrigated by placing water in earthen pots. The water percolates to the roots of the plant. The water requirement of plants varies with the stage of their growth, and this requirement varies from plant to plant. Generally, the plant needs less water when it is young, and when it is shooting, booting and earing, its water need increases. As it approaches maturity, it does not need as much water for further growth. Rainwater is a boon to agriculture. It has to be managed properly. It should be managed where it falls and followed along its flow. Soil erosion caused by runoff water from cultivated fields should be avoided at all costs. For making maximum use of rainwater, it is best to grow crops during the rainy season. This also provides plant cover to protect the soil against erosion. Rainwater gets collected in percolation wells, tanks, farm ponds and check dams. It should be conserved in its place of storage and used judiciously. In some catchment areas of dams, cetyl alcohol is sprayed over the water surface to reduce loss by evaporation, thus conserving water for irrigation. In some other places, loss by evaporation is reduced by shifting water from smaller places to a bigger and deeper reservoir, thus reducing the area of evaporation. The harvested water is recycled for use in agriculture. Sources of irrigation water in our country are limited. The water should therefore be used most judiciously. Conserving water for irrigation is of utmost importance to the farmer and the country. The water thus saved will help in irrigating many more hectares of land, giving additional crop yields, leading to the prosperity of the farmer, his family, and the country.
The Taj Mahal, our monument of pride at Agra. The focus and concern has shifted from freezing its beauty and splendor through a lens or a brush to the critical debates of its becoming pale and corroded. This is but one isolated example of a problem of global proportions. let us move a few hundred kilometers away to Delhi, the capital of India. This is the fourth most polluted city in the world. Perhaps the situation is worse in the top three: Mexico, Rio de Janeiro, and Tokyo. Air pollution indicators far exceed the national environmental standards. Just stay here for a few minutes and you will feel an irritation in your eyes, and smoke blurs the vision.
See, it has been seen in the recent WHO report, which was published in 1992, that 1 out of 8 children, they suffer from some kind of respiratory problem, such as asthma, chronic bronchitis, and upper respiratory symptoms. Recently, a few studies have been published from South California, where they have correlated the respiratory symptoms in various children and their lung function with reference to the levels of these pollutants. And this 10 years, this study, which was followed up over 10 years period, has shown a good correlation between the levels of these pollutants and the respiratory ailments in these children. It is very difficult to fractionate the effect of a single pollutant because all of them are there simultaneously. So, but what we can say that there is definitely a synergistic effect. But in some cases, a particular pollutant has been associated with a particular problem. So these studies are there.
Not only is air pollution injurious to the health of children, even adults and especially senior citizens suffer from its hazards. Shri Prithvi Singh remembers vividly how this city was nearly 50 years ago.
आज मैं इसी गांव का निवासी हूँ। मेरा जन्म यहीं का है। मेरे 60 साल की उम्र से मुझे दिल्ली की काफ़ी अच्छी तरह याद है। मैं यहाँ पर ही रहा, यहीं पढ़ा और मैंने इस दिल्ली को बहुत हरा भरा देखा है। बड़ी खूबसूरत दिल्ली देखी है मैंने। अगर 60 साल पहले की दिल्ली बताई जाए तो प्रदूषण नाम की चीज़ बिल्कुल नहीं थी यहाँ पर। बहुत विदेशी लोग यहाँ पर देखने के लिए बड़ी खुशी से आते थे और आज का तो ये हालत है कि प्रदूषण की वजह से परेशानी ज़्यादा है। सभी दरख्त जो हैं वो कम हो गए हैं।
What we are doing to the environment and to the balance in nature is of grave concern, and we will explore this aspect in detail in the first part of our program, Slow Poisoning. Imagine this basketball to be the planet Earth. Then, the atmosphere is thinner than this sheet of newspaper. Without its atmosphere, the Earth would be as lifeless as the moon and frozen, instead of averaging about 15 degrees Celsius. Our present-day lower atmosphere is a mixture of gases, chiefly 78% nitrogen and 21% oxygen. The remaining roughly 1% includes gases such as carbon dioxide and some traces of other gases, together with a variable amount of water vapor whose concentration may range from a negligibly small value in a desert to about 5% in a steamy forest. The composition of air has not changed fundamentally over the last 600 million years. So when did pollution begin to become a problem? presumably 2 centuries ago in the early 1800s after the first coal-powered industrial revolution. The rapid growth of global population, shifts in lifestyle, large-scale industrial production and automobile exhaust are relatively new contributors. Finding their way into the air, a number of contaminants and industrial discharges aggravate the situation. The smoking stacks, acrid exhaust fumes, and industrial haze once covering only urban towns and cities have now spread to blanket entire regions. There is no longer any room for doubt about the harmful consequences. Statues and buildings have been blackened with soot from unburned fuel, consisting mostly of carbon particles and industrial ashes. When is air clean and when is it polluted? And does natural mean clean? These questions are difficult to answer. Technically, air is never perfectly clean, but is considered polluted when the presence of one or more undesirable contaminants in the atmosphere alter the chemical or physical or biological quality of air. We consider only those impurities as air pollutants which are present in sufficient concentration to cause some measurable adverse effects on either living organisms or on materials, and even if these interfere with the normal enjoyment of life and nature. The source of airborne wastes are so many and so varied that pollution of the atmosphere is a fait accompli. Much of this still happens from uncontrollable natural processes. Like this eruption of a volcano. This volcano is not only devastating in its vicinity, but also releases massive discharges of dust, ashes, and sulfurous gases high into the atmosphere, affecting it for a long period of time. Dust and sand are light enough to be carried by wind in storms like this one. Raging forest fires release quantities of noxious gases and odors in a few hours that the slower, natural processes of decay of surface debris would have taken many years. And these are the pictures of rising pitch-black smoke over Kuwait City during the Iraq-America Gulf War in 1991. Smoke, fumes, and aerosols are extremely fine particles. Their sizes are less than a micrometer, 1,000 times smaller than a grain of sand. Larger solid particles temporarily suspended in air are commonly called dust. Solid particles like this coal dust in a mine tunnel are less than 10 micrometers in size. When they become airborne, they stay there suspended for a long time before settling to the ground. They are called suspended particulate matter, or just SPM, or PM10 by workers in air pollution. Power stations and industries release as much as up to 75% of SPM into the atmosphere. Some are as small as just 2.5 micrometers. And there is increasing scientific and medical evidence that exposure to these ultra-fine particulate matter causes and aggravates respiratory diseases and can even cause cancer. In recent years, the atmosphere's burden of pollutants has increased dramatically. The burning of fuels such as coal, petrol, diesel, LPG and so on is still the main source of civilized air pollution because the steps to remove the byproducts that result from their combustion are inadequate. If carbon dioxide and water vapor were the only products, the burning of fossil fuels would not pollute the air. But the circumstances are not so ideal. The combustion produces other products, including small amounts of chemicals like sulfur and nitrogen. More often than not, incomplete combustion of coal, diesel and petrol in inefficient engines produces carbon monoxide instead of carbon dioxide. Although carbon monoxide is quickly removed from the atmosphere, it can nevertheless be highly toxic. Normally, oxygen atoms attach themselves to hemoglobin in the blood, forming oxyhemoglobin, allowing the transport of oxygen throughout the body. Carbon monoxide can, very similarly, combine with hemoglobin, and when this happens, it effectively shuts out the possibility of oxygen attaching to hemoglobin. Carbon monoxide causes poisoning symptoms depending on how much of the blood's hemoglobin it attaches to. When this happens, people may suffer from headache, dizziness, heart and lung problems, and in severe cases, even death. The addition of pollutants to the atmosphere can have a significant global impact, as in the now infamous ozone hole case. The outer reaches of the atmosphere, near the stratopause, contains a layer of ozone. The thickness of the layer varies from 10 kilometers to as high as 50 kilometers. Ozone splits into oxygen when hit by ultraviolet radiation. The energy of the life-damaging UV radiation is thus converted into harmless heat. Oxygen reconverts into ozone, thereby maintaining the ozone levels. Hence, the ozone layer functions as a global shield from the solar ultraviolet radiation. But this ozone screen is under attack. Scientists believe chemicals, particularly widely used chlorofluorocarbons or CFCs, have thinned the protective ozone around the globe. This damage is now taken most seriously. When CFCs were first introduced in 1930s, it was called a miracle gas because of its non-poisonous, non-inflammable and immensely stable nature. Foams and aerosol sprays used CFCs as propellants. CFCs are also used in making foam for mattresses and curtains, and packaging materials for insulation. When ultraviolet radiations strike them, a CFC molecule releases a chlorine atom. Then, the free chlorine atom steals one oxygen atom from triatomic ozone, thus destroying it and forming chlorine monoxide and diatomic oxygen molecule. But the oxygen atom in chlorine monoxide is attracted to the free oxygen atom and breaks away to form an ordinary oxygen molecule. The chlorine atom is left intact to continue the catalytic damage of ozone for almost 100 years. Every % reduction of this protective ozone screen could cause anything up to a 3% increase in the amount of ultraviolet radiations reaching the surface of the Earth, and this can cause a great deal of damage. Since 1980, we have experienced the 13 hottest years on record in just 20 years. Is our world getting warmer? Yes, scientists believe that global warming is real. The temperature on Earth is already 1 degree Celsius higher than the last century. There may be many causes for this variation in global temperature, but some scientists believe that this intensification of the greenhouse effect is one of the reasons. The incoming solar radiant energy strikes Earth's surface, causing it to emit long waves of infrared radiations like a giant radiator. Because of the atmosphere, however, only a fraction of that heat makes it directly into space. The rest is trapped in the lower atmosphere and absorbed by the gases. This entire process of temporary retention of heat is what is called greenhouse effect. And because of this effect, the global climate is much warmer than it would be otherwise. And evidence suggests that there is a discernible human influence on global climate. Scientists on Hawaii's Mauna Loa have recorded a steady increase in the concentration of carbon dioxide for 32 years. And it is now about 30% higher than it was before the Industrial Revolution. With increased heating, more water evaporates from oceans, lakes, and soils. Because a warmer atmosphere can hold more water vapor, this creates a power feedback loop. The hotter it gets, the higher the water vapor content of air, and thus greater the warming. And we have little direct control over the volume of water in the atmosphere. Adding to the misery, we produce other greenhouse gases too, which intensify the effect. Generated by bacteria in paddy fields, decomposing garbage, guts of ruminant cattle ranching, and incomplete burning. Methane is the principal gaseous ingredient of a wide array of volatile organic compounds. Methane persists in the atmosphere for nearly a decade and absorbs 20 to 30 times more heat than carbon dioxide. The potency of methane has contributed an estimated 15 to 20% to the global warming in modern times. nitrous oxide, better known as laughing gas, plays a somber role in the atmosphere. Produced by microbes in soil, its increase comes from nitrogen fertilizers in agriculture, and slash and burn farming, as well as by fossil fuel emissions in industries. Although nitrous oxide is roughly 200 times as heat absorbent, it adds about 5% to the man-made burden of the atmosphere warming. If this trend of increasing concentration of greenhouse gases in the atmosphere continues, there would be warming of atmosphere. We appear to have opened up a Pandora's box. Let us look up the recipe of doom that we appear to have created. The Industrial Revolution, in a way, heralded the beginning. For the first time, large amounts of pollutants started getting into the atmosphere. As we progressed, we continued to add more and more to this problem. Automobiles, larger and faster transport systems, all these only ended up in adding newer materials into the atmosphere. Our understanding of the harmful effects of our actions simply doesn't appear to be keeping pace. Pollutants can have a range of effects on the human body, ranging from a simple urge to cough or watering of the eyes to asthma, asphyxiation, and cancer. Plants and animals also have to pay for our folly. Large-scale damage in the form of acid rain, ozone hole, and greenhouse warming of the earth can also occur. It is almost as if we are sitting on a time bomb waiting for us to make just that wrong move. The Bhopal gas tragedy is a grim reminder we dare not forget. The logical question to ask would be, so what can we do about it? Rolling back, of course, is impossible. Lessening our greed and increasing our awareness and sensitivity could help. We will look at some such measures in the next part. Till then, goodbye and take care.
Today's children, growing up in cities, are isolated from nature. They have no idea that just outside the classroom window, the beautiful, diverse world of nature awaits them. We just have to open their eyes. There are so many different plants and animals, and all of them are connected in a complex web of life that begins with the sun. Plants use the sun's energy and mineral nutrients in the soil to make food. This is the basic food for the entire animal kingdom. So, you have small insects that feed on these plants. You have fishes that feed on water plants. Birds that feed on fishes and frogs. Snakes and lizards which form the reptiles. And finally, the large mammals, tigers, lions, the carnivores, who ultimately feed on all the lower animals. And then, there is us. We too are part of the web of life. As children learn more about nature, they understand more about themselves. All of us are connected to every living thing on Earth. Nature walks can help children discover that.
No, Priya's taking. Only two, no?
No, there's so many. Actually, can we take this back and just go and see what it is? Huh?
I'll catch it in my hand.
Okay, let's just go back and see what this is, okay? We'll take it like this. When we go out for these walks and everything, sometimes, I too don't know much about insects. To be frank, I have to also go and look up and find out the names and all that. But I am able to tell them a little bit about what they see. And they're very observant. Even the little crevices and all, and little between the grass which I can't look for, the insects and all, they'll get there. Even the tiny eggs and everything. They just like to be out, Even the classes which you take and all, you can just sit out and talk. It's like rather discussion, it's a dialogue between us. Not that I have a book and I say, okay, let's go and see this. See, there's nothing specific nature walk in the sense, they're just observing the nature. You can't be specific and say you go and look for what. It's not like if they find something, they come and ask you, like, Auntie, what's this, what have I found? And then maybe we all get together, sit down there and discuss what we have found.
We see nature, nature, and we see nests, and we hear birds. And we see trees.
They know that a tree also has life, and you can actually find a child going and hugging a tree and really feeling for it. And they don't pluck any flowers. They feel for it, and you don't even find a child kicking a caterpillar off the way or something like that. They pick it up and put it at the side. And there's some of them who are so sensitive that they say, oh, don't stamp the grass. So the other one says, where should I walk then,
Sometimes we also climb trees. Yeah.
What happened?
Yuck.
What is it?
It's a cross sitting up there.
It develops the senses, you know, that's one thing I've noticed when you walk. You got to be alert, you listen to the sound, you smell, you can feel. And the children, I think, should develop their senses.
After a nature walk, it's time for the children to remember their adventures by recording their findings and sharing them with each other.
This one. Which one did you see? This one. Okay, and I'll tell you about that egret. It's called large egret. What is it called? Large egret.
The teacher, through books and pictures, can help the class learn about what they've seen. And the children can give their impressions too. Let nature enter a child's imagination, and it can express itself in many ways. Leaf rubbing is a simple activity. Just lay a leaf under paper and rub a crayon over it. leaving a colorful impression. This is also a fun way for children to learn about different types of trees and plants. The teacher can join in too, making posters to explain more difficult concepts. Assign a report on a specific creature. Let the child imagine. Just what is a bee? An aquarium is a complete world in itself. The fish feed on water plants, which grow in gravel. And snails keep the glass clean as they feed. This self-sufficient ecosystem can be enjoyed by the class throughout the year. Classroom activities create a better understanding of the world of nature.
Those are these large egrets. But the cattle egret that you remember, we said
If children have the freedom to express themselves, this world will brighten the classroom and the child's imagination.
They're very noisy. Generally, they're very noisy. And you know, some go crazy here and there. But we just don't generally control them. And it's all in the learning process. Whatever they do, you learn with the kids. And you know, the subtle things which you may really miss, you know, you really get it from the kid. We went there with the kids to the lake. And they were watching the ripples. And the movement of the ripples, you know, one child immediately said, aren't our thoughts also equivalent to the movement of the ripples, auntie? You know, immediately, you know, he's watching the ripples and he's able to relate it to the thoughts, a second standard child.
Suppose we'll be learning about a snail. Then we go looking only for snails. And we collect those shells, or if we actually find one, we sit and observe it and see how it moves around and where the feelers and the eyes of the snail and the breathing hole and all that stuff. We get back, pick up one and bring him to class and study the behavior of the snail, what it eats and everything. And then they write about it, maybe on a worksheet or in their book, whatever. Even that's not copied from the board. They write what they have understood and what they feel like writing, how they express themselves. And it's not always like facts, because sometimes they write, for creative writing, we do something like, I am a snail.
During playtime, children can play a game based on the food chain. Each child can choose an animal and experience the web of life for themselves. By becoming an animal, children will have a better understanding of the cycle of life in nature.
Food chain is a type of animal. Food chain is like how one person eats the other person eats the other person. So it's called a chain, and the food. So the animal eats the food, and it becomes a long chain. Like the chain is like very long. It's a very long chain. But I don't know that the snakes will eat.
In a
the
way,
frogs
man
and
is the strangest creature of them all. Only we have the power to destroy nature or to save it. Nature walks can help children to discover that they are connected to every living thing on the earth and create a lifelong love and respect for nature.
दरवाजा ठीक से बंद कर लेना बेटी।
काश खुले आकाश को मैं छू पाती। पर पंख कहाँ है मेरे? मैं क्या कर सकती हूँ?
मैं बचपन से यही चाहती थी कि मैं सब ऐसे काम करूं जो लोग समझते हैं सिर्फ लड़के कर सकते हैं। ठीक है, अगर लड़की है तो क्या हुआ, ऐसा कोई काम नहीं है दुनिया में जो लड़के सिर्फ कर सकते हैं और लड़कियां नहीं कर सकती। शुरुआत से ही मुझे शौक था कि मैं पायलट बनूं, उड़ान करूं, हवाई जहाज़ उड़ाऊं, और मैंने ये करके दिखाया। ठीक है, कठिनाइयाँ आती हैं, मुश्किलें आती हैं, लेकिन ऐसी कोई बाधा नहीं है जो आप इसलिए नहीं बन सकते पायलट क्योंकि आप लड़की हो। ऐसा कोई काम नहीं है जो लड़की नहीं कर सकती। ठीक है, जो मुश्किलें आती हैं वो सामाजिक वातावरण की वजह से हैं, लेकिन इस वजह से नहीं है क्योंकि लड़की है। आजकल लड़कियां तो कितनी दिशाओं में कहां कहां काम कर रही हैं, साइंटिस्ट हैं, इंजीनियर्स हैं, डॉक्टर्स हैं, बल्कि आजकल स्पेस में भी हैं। कोई भी लड़की पायलट बनना चाहे तो बन सकती है। और अगर आपको आत्मविश्वास अपना बढ़ाना है तो अपने आप पर ज़्यादा भरोसा कीजिए, आत्मविश्वास बढ़ाइए और ये मत समझिए कि आप किसी से कम हैं। आप सब कुछ कर सकते हैं जो भी लड़के करते हैं। ये तो एक छोटा सा जहाज़ है जो शुरुआत में लोग सीखने के लिए उड़ाना शुरू करते हैं, इसके अंदर खिलौने की तरह है। मैं तो इससे बड़ा भी जहाज़ उड़ा सकती हूँ, और मैं ही नहीं, आप भी कर सकते हैं।
हॉर्स राइडिंग लेने का मेरा बचपन से मेरा शौक़ है कि मैं बड़ी होकर राइडिंग सीखूं। और लड़की होने का सवाल तो ऐसा कोई है नहीं हमारे में के लिए लड़का है तो ये करेगा, ये लड़की नहीं करेगी। इक्वलिटी है, लड़का लड़की का तो कुछ सवाल ही नहीं उठता।
आई एम द ऑल इंडिया बेस्ट कैडेट और मेरे को बहुत अच्छा लगता है क्योंकि मैंने बहुत कष्ट उठाए थे, इसलिए मेरा पूरा साल इसमें गया था और मैं जब बड़ी होंगी तब मेरे को डॉक्टर बनना है।
Injury in the joint, then what type of bandage you will apply here?
You can apply physical of tape bandage.
Can you?
मेरे मन में ये भावना है और पक्का विश्वास है कि लड़की को पूरी अपॉर्चुनिटीज़ जैसे लड़के को दी जाए, जैसे लड़के को अपॉर्चुनिटी मिलती है, इसी प्रकार लड़की को अपॉर्चुनिटी अगर मिल जाए और उस अपॉर्चुनिटी का वो फ़ायदा उठा ले तो वो किसी से कम नहीं है। वो अपने भाई से कम नहीं है, अपने कोलीग से कम नहीं है। लेकिन प्रॉब्लम ये है उसको अपॉर्चुनिटी मिले कि नहीं मिले, और अगर मिल जाए और उसका वो फ़ायदा ना उठाए तो फिर वो अपना नुकसान खुद करती है। एक विश्वास से वो चले कि जो मुझे अपॉर्चुनिटी मिली है मैं इसमें पूरी मेहनत और लगन से काम करूं और मैं सीखूं और अपने पाँव पे पहले खड़ी हूं। अगर उसका ये लक्ष्य हो कि मैंने अपने पाँव पे पहले खड़े होना जैसे मेरा भाई खड़ा होता है, मैं अपने माँ बाप के लिए वैसे ही कमाऊ बनूं जैसे मेरा भाई बनेगा, मैं अपने माँ बाप का नाम वैसे ही आगे लेके जाऊं जैसे मेरा भाई लेके जाएगा, तो कोई बात नहीं है वो किसी से पीछे रह जाए। लड़की किसी से कम नहीं है, ये सिर्फ उसकी मानसिक बात है। अगर वो अपने आप को सोचे कि मैं कमजोर हूं तो उसको कोई बहादुर नहीं बना सकता, लेकिन वो सोचे कि मैं बहादुर हूं और साथ में बहादुरी के लिए अपनी मेहनत करे, लगन से मेहनत करे, तो वो किसी से कम नहीं है।
ये सब चेहरे तो मेरे ही जैसे हैं। अपना रास्ता मुझे खुद तय करना होगा, मुझे खुद उड़ान लेनी होगी। पर पंख कहाँ है मेरे? है ना, अपने ऊपर मेरा अपना विश्वास। इसी विश्वास के सहारे मुझे भी छूना है आकाश।
मित्रों, आपको हमारे कार्यक्रम कैसे लगे? आप अपनी प्रतिक्रियाएं और सुझाव हमें भेज सकते हैं। हमारा पता है किशोर मंच, CIET NCERT, श्री अरविंदो मार्ग, नई दिल्ली 110016। आप हमें ईमेल भी कर सकते हैं। हमारा ईमेल पता है ciet.kishormanch@gmail.com और हमारे टोल फ्री नंबर हैं 1800111265 और 1800112199।